Good morning, everyone. It's really nice to see everyone in person. I have a lot of friends, a lot of colleagues, we think a lot on Zoom, and the first time we got real world evidence that we're actually real people, not just bots. And so, welcome to 2023 IDES Annual Symposium. My name is Chen, and I'm a professor of operations management and business analytics. At the Sheridan School, I'm also serving on the executive committee of IDES. Now, it gives me a lot of pleasure to introduce our one and only Dr. Alexis, the founding director of IDES and the SSEC, global distinguished professor who was recently elected to National Academy of the Sciences, and he's also professor of computer science, in addition to being professor of physics and astronomy. So now Alex will give us welcome opening remarks. Thank you.
So thank you very much for the introduction. So this is now, I've even lost count of how many years we were starting. So it was something like 15 or 16 years ago that IDES was founded. And this year is very special. So finally, as in data science and AI is becoming a central piece of Hopkins and Hopkins' future, and it looks like that this will be an unprecedented growth in the history of the university. So I will try to shout through the noise, but so there will be more than 100 faculty positions dedicated to data science and AI in the next 10 years. So recognizing this, we have been planning how can we ensure that IDES can help to get this effort jump-started. So for the last 2 years, we have been in close discussions with the Emerging AIX project. And as a result, IDES, which was founded in arts and sciences, is now going to move over to the engineering school, which is going to be the center of this data science effort. But we will continue to engage with all the schools across the university, as we have done in the past. So AI is going to change science, and it is going to change the university. But it is important to understand that AI is not just about purely deep learning and running some of these frameworks and buying many GPUs. It is also about how can we use data in very unique ways. And it goes in science, it will go even beyond this. So right now we think about AI as a new way to analyze existing data and find new patterns in these data sets. But actually, at the same time, what is going on in society, we are trusting AI to drive our cars, or pretty soon we will. And why cannot it drive our microscopes and our telescopes? So we are getting too good in collecting data in many ways. So today, there is a new kind of science emerging, something between the individual PI grants of $300,000 and the $1 billion national laboratories. I call this mid-scale science, where people so a couple of faculty get together across a few universities, get a $20 million grant, build a new kind of instrument, and then with a small group, using very advanced data acquisition techniques, they can easily collect petabytes of data in a year. And we haven't quite figured out what to do with this data. But this mid-scale science, those projects are probably representing for the next few decades the sweet spot of science in the world. Because they are not going to spend too many dollars, they don't want to create our own computer center like a national laboratory. But they can do incredibly ambitious projects. But the question is that how can they make the science spending even more efficient? And what is starting to emerge is that today's trend is that we are getting good at collecting data, and we gobble up a lot of data and think about it afterwards, what to do with it, and how can we analyze it. And so why can't we actually then use the AI techniques to focus our experiments to collect more data that is relevant to the best science that we want to do? And I think this is a completely untouched area to essentially hand over more of the control to the AI systems in our whole scientific process. But this requires also heavy thinking. How can we translate essentially our science goals into a cost function that can be tackled in an algorithmic fashion? And how can we also train the next generation? So there is kind of a chaotic situation in science today. We try to figure out our place in the world. So there are the big 3 or 4 companies, Google, Facebook, Microsoft, Amazon, who are spending, and OpenAI, who are spending enormous amounts of money in training AI systems. And the question is, how can a university be competitive in this world? Also, the agility of these companies is unimaginable at the university scale, where they kind of go from concept to prototype to actually completed systems in a matter of months, where it takes us 6 months to talk to the dean at the university. So we have to become much more agile and also figure out with a razor-sharp focus that what are the areas where universities can make a creative and significant contribution to this. And in many ways, we will not still, no matter what we do, we will not be able to compete on the scale of the hardware that is being deployed. But where we can compete is to have innovative ideas in the mathematics of deep learning. Also for science, we will need to figure out how can we use AI in a way that we can understand what is happening inside the neural nets. How can we do proper statistical inference instead of using a black box? And so when we had the symposium in the spring, then Andrew Moore, actually, who used to run the AI infrastructure for Google, mentioned that to the audience that he thinks that the next breakthroughs are going to come from somebody, an assistant professor sitting today in the audience, which was, I think, very encouraging. So that he also sees that the future inventions are very likely coming from independent thinkers in academia, who instead of being part of the mad rush forward in the industry have actually a little more time to think. And I think, so in this spirit, a lot of the talks today are really dedicated to interesting uses of AI. And so with this sets the tone, I think, of this meeting today, and probably for our research directors for the next decade. And so I would like to introduce our first speaker. So Alison Vettel, she's a Bloomberg professor, and she's also the associate director of the IDES project. And she's also the director of the Lawson Center, and she's going to talk about the relation of AI and genomics. Thank you.
Thank you very much for that introduction. So yeah, thank you, Alex, for that great introduction. I'm really excited to be joining you today. As he said, I'm an associate professor in biomedical engineering. Alright, thank you very much. So yeah, thanks for everyone for joining this morning. So I'm going to be talking about a particular application of machine learning with the type of data that I work with, which is focused on human genetics, and specifically trying to understand the genetics of human disease. So as you probably have heard, or if you do work in this area yourself, you know that we are now able to regularly sequence the full genome of individuals, and this includes patients in clinic and also just commercial applications. You can go and order your whole genome from companies that will happily sell it to you for about $1,000 these days. So it's readily available, it's become significantly more affordable, and with this comes the promise that in theory you could take your full genome sequence and look at it to understand your risk of heritable disease. And if you have a patient who has a suspected genetic disorder, you can use their whole genome to hopefully identify the genetic cause of that disorder that they're experiencing. So this is the hope, and we are now able to collect this data relatively easily and relatively affordably. So I'm going to give you just a very little bit of background to start to get into why this is still a very hard problem. So the standard method for understanding the genetic basis of disease doesn't focus on the variants that we find in whole genome sequencing. It focuses on what we call common variants. So these are changes in DNA that we see frequently in the population. So for example, if you were to look at the genotype of an individual at a particular location, we know that we can abbreviate your genome sequence with the A's, C's, T's, and G's that we find. So at a particular locus, maybe about half of you in the room have the usual, the common genotype of AA, but some people are carrying a C at that location, and they have either a heterozygous CA or a homozygous CC. Now, the standard way to understand genetics of disease is to do these large-scale association studies where you collect a number of individuals with the disease and a number without, cases and controls, and you run very simple statistical tests to look for association between a particular genotype and risk of a particular disease. So you might find that people who carry a particular genotype at a given locus have an increased risk of having disease. You can assign a p-value to this and an odds ratio, etc. But this relies on many, many people having that genetic difference in their genome. So what's really challenging is that in many cases we want to understand the consequences of rare variants. So all of us have variants in our genome that we've never seen before in any sequencing study. And this sort of standard association testing quite clearly fails in that case. If we have a patient that has a variant we've never seen before, we cannot simply rely on the results of these association studies to tell us how it changes their risk of any given disease. So what do we do with these rare variants that make each individual unique, where standard association methods simply can't be applied? And complicating this further is that most of the genome doesn't actually have an obvious function at all. So you're probably used to seeing in the news, we identify genes that increase the risk of schizophrenia or genes that increase the risk of diabetes. But these are usually referring to actual genes, which are very small stretches of DNA that encode the instructions for your cell to make a particular protein. Now, these genes are relatively well studied. We understand the function of many of them. We can look at the genetic code to identify the variant within a gene is actually going to change an amino acid that's part of that protein, and therefore potentially change the protein structure. But genes actually only make up about 2% of your DNA sequence. So 98% of your genome is what we call non-coding, or the sequence does not encode for any protein. So for most of your DNA, and therefore for most of the sequence variants that you might have, it's very, very hard to predict their consequences. So what I'm showing here is the variant, someone has a C we've never seen before out in the middle of an intergenic region. It's very hard to guess as to what its consequences might be, whether it has any consequences. If so, what are they? It can be quite difficult to predict this. There are no sort of standard methods that can do this reliably. So not only can we not run association tests, we also can't just look at what we know about the genome and predict what these variants might do. So just to give you some context about how much rare variation we actually see and why this is important, rare variation is actually very, very abundant in the human genome in all of us and is mostly uncharacterized. So each individual, and this is from a reference of healthy individuals, has about 50,000 variants in their genome, so 50,000 places where they're different from most people, where these variants are rare, and here I'm defining it as seen in fewer than 1 in 100 human genome sequences. In addition to these 50,000 that are moderately rare, we have within those thousands of variants that we have never seen before, and a subset of those are likely to be large high-effect variants. So most of these rare variants are non-coding, so they fall into that 98% that don't lie within a gene, and their impact is unknown, so it's not amenable to standard association matters, although I think it underestimates the impact, is the statistic that somewhere around 5% of Americans have a rare genetic disorder. About half of these individuals who have a rare genetic disease do not receive a genetic diagnosis with current methods. So standard methods which look at the exome, which is the region directly in or around those genes that I mentioned, and look at variants that are known or very likely to disrupt those genes. These methods that are currently used in clinic by default do not diagnose about half of these 5% of individuals who have a rare genetic disorder. And I think this is a huge underestimate of the importance of rare genetic variants in health, because not only do they cause these really extreme rare diseases where a patient comes into the clinic, usually a child, and it's clear that they have a genetic disorder, all of us are walking around with rare variants that have sometimes large, sometimes small impacts on our risk of both rare and common disorders. So personal genomics remains actually a very unsolved and challenging problem, despite the advances that we've made in genome sequencing and in our growing understanding of genetics. So the question that I want to talk about today is how can large data and machine learning help us address this problem? So we are interested in building machine learning models from large whole genome sequencing data sets to help us evaluate these rare variants from a new individual genome that we might see in clinic or just our own, for example. And beyond just the whole genome sequencing, what I'm really going to focus on today is the use of other data types, so really diverse multimodal data to help us identify when a patient has disruptive biology in their cells. And so this is going to rely on other sequencing technologies and other high-throughput data sources, including RNA sequencing, epigenetic data, proteomics, and more, to complement the whole genome sequencing that I've been discussing so far. So luckily, there is an explosion, as these prices have increased and technologies have been developed, there's an explosion in the availability not only of whole genome sequencing, but other data types as well. So as genome sequencing has become cheaper and more ubiquitous, we now have millions of genomes being sequenced as part of projects such as UK Biobank and the All of Us effort that is going on. So we're getting hundreds of thousands, now millions of whole genomes, but we're also getting these additional data types that I mentioned. So sequencing can be applied to RNA in the cell to give us estimates of how much each gene is expressed. We can apply it to get estimates of epigenetic changes in the genome. We're now able to measure these both DNA and functional data such as RNA and epigenetics at a single cell and a spatial level in different tissues. So we're really getting this, this is just a very small set of the data types that are available now, but we're really getting very large amounts of data of these diverse types, which I think is very, very exciting, but they don't immediately give us a readout of what these genetic variants are doing. So sophisticated computational methods are really critical to the interpretation of data that has itself become increasingly complex. So I think there's this, thought that we measure things at a more fine-grained and accurate resolution, such as with single-cell RNA sequencing or with spatial transcriptomics. But those of you in the room can probably understand that as data get more complex, actually the computational methods need to be more sophisticated as well. They don't just give us a more precise readout of exactly what we need to know. So how is this useful in understanding the impact of rare genetic variation? So a very simple hypothesis that we and others have explored starts with just the basic idea that if a genetic change in your genome is causing phenotypic or disease impact, it's probably also causing a disruption at a cellular level that we can read out. So in the case of rare non-coding variation, for those of you who don't spend your time thinking about this every day, a non-coding variant that's not within the gene itself often functions through changing when, how much, or where a gene is expressed. So this variant that's between in an intergenic region may affect when one of these two genes nearby is expressed in what cells at what time in response to what environmental changes. So they're what we call regulatory. So the idea is that much of this non-coding variation is likely to result in unusual expression of nearby genes. So the simple approach that we've taken and others have also been looking at is to identify individuals whose gene expression, so the amount of a particular gene that they produce in their cells in the form of RNA, when their expression levels is very far from the population average. So this does rely on large data availability in the sense that we need to build a reference distribution of what normal people look like in terms of their gene expression of each gene. So just to display this we can look at what the sort of normal range of gene expression is for a given gene, one particular protein coding region of the genome, the RNA that is the amount of RNA that's normally produced, perhaps in a given tissue type. And then we may find individuals who are extreme underexpression outliers, so people who barely express this gene compared to the normal population, and individuals who express far more than the normal population. And in some cases, we will find that if you look at the genomes of these outlier individuals, they in fact will have a rare variant near the gene where they are extreme outliers. So that was sort of the basic hypothesis that we began with. So I'll start talking about the results from particular data sets as I go, but to introduce each one, first, one of the data sets that we focus on quite a bit is from the GTEx project. So the GTEx project is looking at genetic effects on gene expression across diverse tissues in the human body. So the most recent release, the rare variant data that we looked at, was from whole genome sequencing of 714 individuals, where we also had access to post-mortem RNA sequencing for a very large number of tissues. So in fact, 54 tissues are available in the most recent release of the GTEx data. And this includes multiple different brain regions, blood, liver, lung, etc. Because it's post-mortem, we have access to this very large number of tissues. And within the GTEx data, we then looked, so we were able to use this set of individuals, about 714, to build reference distributions for what gene expression normally looks like in each tissue across this large set of individuals, and therefore identify individuals who deviate from that normal expression pattern significantly for any given gene. And when we look at these individuals who are outliers in RNA expression for any gene, and then look nearby that gene in their whole genome sequencing to see what variants they harbor, we do find that individuals who are overexpression outliers and underexpression outliers, especially the extremes, do tend to have rare genetic variants nearby those genes of types that are, in fact, likely to produce the functional effect that we see, such as individuals with duplications of a nearby region are likely to be overexpression outliers, individuals with deletions, severe coding variants, etc., are more likely to be underexpression outliers, but in fact, many of the individuals who have really extreme changes in their gene expression do in fact harbor a genetic variant that is likely to be causing it. But this is just an enrichment analysis at this point. While it does rely on looking at the distribution across many individuals, I haven't yet told you about the machine learning part. I will note, for those of you who are interested in genomics, we look at a lot of changes in RNA that are not just expression, including gene splicing, allele-specific expression, and others, and see similar patterns there. But we did want to see if we could put this together into a system that could actually give us a prediction of the probability that any given variant, a rare variant in a new individual, does have an effect on this person's cellular biology and therefore potentially on their health. And the hypothesis, again, based on our original idea, is that a rare variant that is impacting their health should also have a molecular signature. So in addition to looking at their whole genome sequencing, if you have in the individual patient or person that you're looking at functional data such as RNA sequencing, protein levels, epigenetic data, etc., we would like to combine that into a unified framework to predict which genetic variants in that individual's genome are most likely to be functional. So we developed a method called Watershed, which integrates personal whole genome sequencing along with other functional data such as RNA sequencing. So this is just a probabilistic graphical model that we developed that has sort of three core components. One is what I'm calling G. These are the features of that individual's whole genome sequence. So that can include things like how many variants they have near a given gene, how far away they are from the gene itself, whether those variants are in regions that are conserved across species or have other evidence of selective constraint on those regions. In fact, a lot is being hidden in that variable G. We use actually the output of machine learning models, including deep learning models based on the sequence itself of what is the likely consequences of this region. But I think the key here is that we're able to integrate other data types. So the bottom layer, skipping all the way to the bottom E, are measurements obtained from other functional data such as RNA sequencing, protein, etc., that tell us what it looks like is happening in these individual cells and whether they are different from the usual population. So these variables E are observed variables that tell us whether people have very extreme levels of whatever signal you happen to be looking at that you have available for this patient. Now, the thing that we actually want to predict are these unobserved latent variables Z that we intend to encode whether or not a given rare variant has an effect on any of the measurements E that we're looking at. So this is a model that is easily extensible to any new molecular signature or data type that you have available, as long as you can use that data type to determine whether your individual is extreme compared to the general population. So to just give a little bit more technical detail, the structure of this model is such that essentially this top layer is a generalization of logistic regression. Basically, it's an Ising conditional random field where we assume that the latent variable Z that says whether or not the genetic variant is likely to be functional is predicted based on our observed features of the genome G, but they're also related to each other. And this is relevant for the data types that I'll be talking about because in many cases we have multiple measurements, such as expression across multiple different tissues, which are not independent. So if a variant affects one brain region, it's also likely to affect expression in another brain region, perhaps slightly less likely to affect expression in blood. And so the weights between these variables Z that connect them in this conditional random field are learned also and tell you how close, how likely a variant is to be functional in multiple of these different either tissues, contexts, data types, etc. So a nice thing about our model, particularly in this field of personal genomics, which is very early in understanding which genetic variants are actually high impact and which are not, is that Watershed is trained in an unsupervised manner, meaning that Z is unobserved. We don't require any label training data about which variants actually are functional and which are not. It's quite efficient. Our models are not huge. So we have just this one layer of latent variables. There's usually, definitely less than 100 of these variables Z, so it's relatively efficient to train using expectation maximization. And in the end, we're able to take a new patient, their whole genome sequence, and any subset of additional measurements that are available. They may only have RNA-seq from blood. They may have, a couple of different measurements from skin and blood, etc. But we can use our trained model to score the posterior probability that a variant seen in a new patient is actually impacting their gene regulation. So we again applied this model, Watershed, to the GTEx project data. So we trained a model for Watershed in a couple of different ways, but across all of these different tissues that are available, across all of our individuals. And we're able to show that, in fact, Watershed performs quite a lot better than any existing method that is prioritizing rare variants based on only the whole genome sequencing alone. So adding in the RNA expression levels across whatever tissues are available dramatically improves our ability to identify which genetic variants are actually affecting a new individual. So in this case, it's trained across just RNA sequencing, but across multiple different tissues and across multiple different readouts of RNA-seq, including splicing, allele-specific expression, and then just total expression. So in addition to getting quite a large boost in improvement of predicting which variants are going to affect RNA-seq in the GTEx data itself, we also replicated the predictions in independent data that was collected entirely separately from the GTEx project, identifying individuals who share variants between the two data sets and showing that we do in fact predict their impact in this held out data set. And then went through and actually validated several of the variants effects in cell culture using CRISPR-Cas9 editing. And one thing that I want to highlight here is that Watershed really dramatically improves the identification of rare variants with predicted high functional impact. So the best models that we have available from genome sequencing alone, and this includes predictions that are actually currently being used in clinic for things like variants that are within a coding region that change splicing, etc. The proportion of even the strongest predicted variants from the genome alone that actually lead to dramatic changes in the cell of our patients is lower than 20%. So even the variants that we think have the strongest effect just from genome sequence alone with the best methods that people have developed out there, they actually don't lead to massive changes in individuals' cells with a very high probability at all. However, if we have seen from that individual patient on RNA sequencing or other functional data, our ability to identify variants that actually are extremely likely to lead to, or very highly likely to lead to actual outlier behavior improves dramatically. So Watershed is taking us from a place where, from the genome alone, our predictions are really quite weak, to a place where I think this is much more reliable to use, potentially in clinical practice. So we've, in more recent years, new data is becoming available, and we've begun to apply Watershed in a diverse set of data types and in a bunch of new cohorts. And one of those examples comes from an MD PhD student in my lab, Lee, who has recently applied this extended Watershed to apply to a bunch of new data types in a larger data set from the MESA data, the multi-ethnic study of atherosclerosis, which gives us, in addition to whole genome sequencing for a number of individuals, also for over 1,000 individuals, multi-omic measurements that include methylation, transcriptomics or RNA, and proteomics as well, and actually across multiple time points for some of those same individuals. And we were then able to adapt Watershed to these new data types and to these multiple time points to build a predictive model for these individuals as well. And very simply, we see similar behavior for the MESA cohort across all these different data modalities as we saw in GTEx across different tissues. So we are, in fact, very clearly able to better identify genetic variants that will have effects on methylation, splicing, protein levels, in addition to RNA, compared to a model that's built from the genome, the whole genome sequence and annotations on that alone. So the performance is held up and now applied to this much bigger data set where we're able to look at a much larger range of genetic variants than we were previously from GTEx only. Just very quickly, we also see that there is a benefit to using this conditional random field model because we do in fact see that the weights between these different omic signals, so whether a genetic variant affects methylation or epigenetics, and whether it affects, for instance, RNA or splicing, those are correlated with each other. We do see that cascade of effects across these different levels of gene regulation, and looking at them all together is in fact benefiting the model. So we also see that the variants that are prioritized within MESA, this is a large enough data set that we can actually take the specific variants that were identified in MESA and then cross-reference them in larger data sets of complex traits, in this case, such as height, and see that variants that Watershed predicted would be potentially functional do in fact show higher effect sizes on changing individuals' height. And so we see that for all the data types that we're able to look at, so variants that are predicted to affect RNA, splicing and protein, maybe to a lesser extent methylation, do in fact have a larger impact on height in these very large association studies where we can start to look at some of the variants that have frequencies of like 1% or 0.5%. For time, I'll just quickly tell you about one more application. So this is actually a much smaller data set, the Undiagnosed Disease Network, but it's one I'm particularly excited about for two reasons. One is its actual rare disease families who are in fact not diagnosed by standard methods and are out there hoping that they will get an explanation for the disease that their family member may have, but also because this is an example of applying Watershed to a new data type, which in this case is long-read data. So when we look at the structure of the human genome, we see that it's actually quite complicated, and there's not only changes such as gene splicing that we'd like to be able to look at, but also structural variants, where rather than just an individual nucleotide changing from, say, an A to a C, we're getting large changes such as deleting a whole region, sometimes complicated inversions, duplications, and all sorts of large-scale structural variants that may be quite large. And standard whole genome sequencing, which is based on very short reads of, say, 100 base pairs or so, cannot identify or call these structural variants accurately. But as you can imagine, such large, massive changes to your genome as structural variants represent are potentially even more likely to actually cause functional problems in the cells of these individuals. And so from long-read data, which using nanopore technology now gives us the ability to actually call these variants much more accurately, we can potentially diagnose a number of patients that were not diagnosed with standard exome sequencing or even standard whole genome sequencing alone. And especially in combination with RNA-seq for these same individuals, we're able to get a much better picture. Now, it's still helpful to have these much larger data sets as sort of to build the background distribution of what gene expression or splicing, et cetera, normally looks like in patients, but from this smaller set of individuals from the UDN who have long-read data available, we can call structural variants and cross-reference them much more accurately. So we've extended Watershed to what we call Watershed SE, which includes a bunch of annotations of structural variants in particular and tries to prioritize these. We have actually been able to find what we believe are likely causal variants for some of the individuals in the UDN. We're in the process of looking over many of these in consultation with genetic counselors who are working with this data with us. But we've identified a few that we think are quite promising, and this is just one example of those. Where we have a large deletion in one of the individuals. This would not have been called from whole exome sequencing alone. It was called from the long-read data available. This individual has some rare cardiovascular traits that they're suffering from, and the deletion that we found is in a gene TANGO2, which is a gene with function that is plausibly connected to this phenotype. And as you can see, the genome alone does not give us enough information to identify this with high confidence, but Watershed SB, which is able to look at RNA-seq from the same individual, identifies this as a highly likely functional variant. Finally, we're now working on an application in a much larger data set, going back to sort of standard whole genome sequencing. We're using the UK Biobank, which has recently released a massive proteomic along with whole genome sequencing data set. So the UK Biobank is a very large and has been very valuable resource in genetics, looking at a number of different traits with over 500,000 participants. Out of those 500,000 participants, we now have available over 50,000 individuals for whom proteomic data has been collected using a technology called O-Link to identify protein expression levels of over 1,000 different proteins. And we've now adapted Watershed to this new proteomic data set and applied it to try to identify rare variants from this much larger cohort that might be causing changes in gene expression at the protein level. Very quickly, we see again that River, or this Watershed, but River is a simplified version of that, performs much better than a genome-only model that does not use the proteomic data. And we can actually, from a data set this large, start to capture more complex effects. So I just wanted to highlight the extensibility of Watershed, which before I showed you can predict the effect of a genetic variant on a single gene across multiple different tissues or across multiple different levels of gene regulation, such as RNA and protein. In this case, we're actually able to start looking at, with this number of individuals, not just effects on a single gene, but multi-gene pathway level effects as well. And in this case, we can take two genes that are either known to work together or where we have evidence of co-regulation and put those together into a Watershed model where, again, we have a link between the genetic effects on multiple different proteins instead of on multiple different tissues, for example, and again, get improved performance to identify genetic variants that have broader effects on multiple genes in that individual's genome. So in conclusion, I hope that you'll take away from this that rare genetic variants do tend to coincide with large changes that are measurable in our individuals, such as gene expression, epigenetics, or protein, and that the explosion in both whole genome sequencing and functional sequencing data can power computational methods, such as the one I presented here at Watershed, to improve personal genomics from just having that promise of maybe we can identify the variants that are affecting health in individuals to something that is hopefully much more reliable and comprehensive. I've left out a lot, including how we deal with things like confounders. There's a lot behind this that I didn't have time to go into today, but I'd be happy to chat with anybody about that. But I'll finish by just thanking my lab. I folded here the individuals that were directly leading the projects that I've mentioned, but the lab as a whole is quite collaborative, so I'd like to thank everyone who's been involved, and I'll take a couple questions perhaps. Sure, so Watershed was not the first model that we developed in the lab. The first model was called River, and it did not have this conditional random field. It just had basically like a single chain where we were looking at one gene at a time, or one tissue at a time, or one phenotype at a time. So the biggest boost in performance does come from adding in any functional signal. So we've compared watershed to river, and watershed does give you a boost, especially when you have something like a tissue where maybe you have fewer measurements such as brain, but you can combine it with a tissue where you have many more measurements such as blood. You do tend to get a boost there. But the improvement of watershed over river is smaller than the improvement of river over using the genome alone. Yes?
I'm interested to know to what extent your model can illuminate the basic biology. So, in the ways in which you can probe the causal relationships between, for example, the non-coding genes and the expression.
Yeah. So I guess there's a couple different ways to interrogate the model to look at that. One is just to simply look at the features that watershed or river prioritize, and those tend to coincide with what we see, frankly, just from enrichment analysis, that yes, variants that are closer to the TSS matter more, variants that are, deletions tend to cause underexpression, etc. But we have looked at, for example, in the MESA data, the cascade from what we imagine to be the cascade from epigenetic changes to transcriptional changes to proteomic changes to see if there's evidence that we're actually identifying that cascade. And I would say that it's hard to interpret the results of watershed as making a strong causal claim, but we see the patterns that you would kind of expect, that you're more likely to have a large deviation in methylation. That doesn't always lead to a large deviation in RNA, which doesn't always lead to a large deviation in protein. There's some buffering there, which corresponds to what we've also seen in our own analysis for common variants affecting that cascade. But to make a causal claim for any individual patient or cohort number, watershed is not doing causal inference, but I think you can see the patterns that you would expect from it. Yeah. Okay. Oh, maybe one more quick.
I've got too many questions, but I'll just ask this, which is basically, you've trained on a very large UK data set now.
Yeah.
And I'm wondering if you've looked at another population, particularly they now sequence cancer patients, the tumors themselves, as they progress, whole genome, something like that, where you've taken the trained model and see if it predicts the things that we find out are true of other populations.
Yeah, absolutely. So I guess there's a couple places where we've done this. We have not applied it to cancer, although I'm quite interested in doing so. I think some of the effects would be much larger there than what we see in complex diseases.
Yeah.
The MESA data had multiple time points, and you can see correspondence between what you see at one time point in a patient to what you see at the next time point, but this is again still not as extreme as what you would see in cancer. We can also see transfer across different genetic ancestries. So in the MESA study, there's a lot of different ancestries that are part of that. That was part of the reason for the collection of this multi-ethnic study. And we can see that actually our model watershed performs better across different ancestries than just sort of standard association testing and even common variants, which are sometimes confounded by that. This tends to do a little bit better. But yeah, we have not actually applied this to cancer in the way that you're suggesting, but I think we're very interested in doing so, and I think it would apply there. Yeah. Okay. Thank you very much.
Thank you, Alexis, for that really inspiring and informative keynote talk. Next, I will introduce Dr. Edward Hunter, who is Director of Engineering at SSEC, is that for Scientific Software Engineering Center. Got it right? Okay. So Edward is going to provide an update on SSEC.
over the next few minutes, I'd like to introduce you to the SSEC. To do this, I'll first describe the purpose and origin of the SSEC centers and the parent organization, the Virtual Institute of Scientific Software. I'll talk a little bit about the new research software engineering staff that we've been recruiting. Then I'll talk a little bit about our software development lifecycle, which is a critical technical component that we use. And then I'll introduce a few of our first projects. Hopefully this together will give you a practical sense of the mission of SSEC. So let me start by describing what motivated the creation of SSEC at JHU. To some people, software development is increasingly recognized as a critical bottleneck and a required investment in academic science. And yet there's a kind of a culture of neglect in universities and funding agencies for open source scientific software development and long-term maintenance. In university culture, software development, or at least professional software development, is often considered somewhat ancillary to the scientific mission. On the other hand, in many industrial labs, or in mission-oriented organizations like NASA, this often isn't the case. And scientific software development and maintenance is a priority line item since the scientific mission is recognized to depend critically on supporting it. At NASA, software development and maintenance is a budget line item just like fuel. The difference in research culture may be due to what defines successful outcome between universities and other scientific organizations. In order for NASA to be successful, beyond writing grants and papers, they have to develop and maintain production systems at high levels of assurance and allow them to complete mission successfully. A similar situation occurs in commercial drug discovery settings, where high throughput systems, screening campaigns, are continually optimized to search for the highest quality molecules to investigate. These systems have to operate constantly improve, and have to easily transition to new personnel with no interruption in service. To make matters worse, funding agencies often fail to prioritize critical software infrastructure, and so it is difficult to hire the best-in-class talent in the absence of long-term steady support. Although it's accidental and artificial, this culture of neglect has the effect of slowing some kinds of scientific progress at universities. IDES has, of course, long understood this viewpoint, as is clear from its many successes, and also by the work that's being discussed here today. To help change this situation, Virtual Institute of Scientific Software, or VIS, was established in early 2022 by a 5-year, $40 million gift from the Eric and Lenny Schmidt family to establish a network of scientific software engineering centers, or SSECs, across 4 universities in the US and the UK. The inaugural centers are located at the University of Cambridge in the UK, the Georgia Institute of Technology, the University of Washington at Seattle, and here at Johns Hopkins. The mission of the VIS and each of the SSEC centers is to accelerate the pace of science by introducing professional research software engineering principles into the academic scientific process. The VIS support allows each center to hire 5 to 6 RSEs with long-term stable funding that is not contingent on a particular grant outcome. RSEs are envisioned to be dual software and science domain specialists who work on software to advance academic research programs. They are envisioned like at-large developers for a department or a school, providing services and collaboration to many science fields. Initially, this and the SSEC centers are supporting researchers already affiliated with Schmidt's Futures programs, such as the Schmidt Science Fellows, whose science is highly promising but blocked by sufficient access to software development resources. In the future, this model of project selection will likely evolve. A big part of setting up SSEC at JHU is defining who the technical staff are within the university organizationally and developing a recruiting strategy around that. To do this, a new pair of research software engineers and data scientists career tracks has been established at JHU. These tracks are designed to be similar to a typical academic style career ladder, such as research scientist. So they break down into assistant, associate, and senior levels, and there's a principal level for distinguished contributions. Since RSEs and data scientists typically work on small teams, we expect some management responsibility at the senior level And this would typically be things like leading and managing project teams, mentoring junior RSEs, and proposing technical approaches and milestone criteria. The creation of these staff lines was driven by the AIX initiative here, which will be using them and staffing its own RSE capability in the near future. Industry experience is considered critical at the outset of SSEC to establish at the university a culture of best practices around software development. These are things like peer code reviews, test-driven development, continuous integration, continuous deployment, and DevOps workflows, software development life cycles and methodologies, excellence in documentation, both written and in code, and so on. Because mission success in industry is long dependent on these, they have a rich history and are widely adopted there, and those practices will greatly enhance the quality and maintainability of software output in a university setting. Our minimum requirement is for 3 to 5 years industry experience, but the initial RSEs at SSEC we've hired so far are all at the senior level with between 10 and 20 years of experience. They come from a mixture of backgrounds. Industry backgrounds include companies like Amazon, Microsoft, the graphics company Vectorworks, and various lesser well-known companies and startups. From the government side, we have backgrounds from the Space Telescope Science Institute here on campus, and major $100 million plus NIH and NSF technology development initiatives. RSEs are considered dual science and software development specialists. 6 of 8 of our RSEs hold PhDs, and we look for people that have experience in 1 or more areas of scientifically strategic importance. Some of the areas the team has expertise in are listed here, including AI/ML, automated microscopy, data modeling, computer vision, NLP, and others. We have built production systems in each of these areas, and in most cases, those are commercial systems. The project teams themselves are the properties shown on the right. They're small, typically between 1/2 and 3 FTEs, but they always have multiple headcounts to ensure robust design and review processes. They are self-organizing, so the team itself determines how to break down project backlog and assign work. They are ephemeral, so the teams form and disband with project lifespans, which is typically between 6 months and a year. And the teams include the science PI and potentially other members of the PI's team. And we internally identify a project lead or RSE responsible for guiding the project. And the teams follow agile development principles that it determines according to its own scheme. But one of the important practices we take from industry is to adapt to the university research setting or software development processes, which aim to make development more predictable with better and quicker outcomes. Development processes are an art form. There's a vast range of possibilities and no best or one-size-fits-all approach. Many possibilities suitable for different industry segments would be too rigid or elaborate for us. So there really isn't a good single existing method to adopt. And instead, we've crafted our own bespoke approach by taking a few best elements from existing methods that we have experience with and we think are lightweight and flexible enough for our purposes while achieving the intended benefits. For us at the moment, this means the rational unified process and the various agile methodologies. From rational, we take the concept of thematic project phases. Each project goes through the same 4 phases: inception, elaboration, construction, and transition. The emphasis on each phase is different, and the graduation between project phases is controlled by predefined milestone criteria. So, for example, to progress from the elaboration phase to the construction phase, we must fulfill the lifecycle architecture milestone, and this involves developing critical prototypes, developing an executable baseline architecture, and demonstrating project risk reduction. Other milestones involve defining scope, implementing scope, and project transition to operations. This is another more schematic view of the development process that we use. Hopefully, this helps to solidify the picture a little bit more for you. Each one of the cycles is a single, approximately 1 month long agile sprint planned by the team immediately prior to execution. Time-box sprints and just-in-time planning are hallmarks of successful agile methods, and they can work very well in an academic setting. The boxes give the milestones with the relevant criteria written underneath. Test-driven development generally begins following LCA, and integration testing with pre-release systems and demos typically happens during construction, potentially around the time of the alpha release. Inception is a little bit special. Little or no coding is happening there. Inception is where we scope the system capabilities and use cases and validate that with the PI and develop at least one candidate approach and behavior that we think is good. Our philosophy about Inception is to be rather intensive. We read all the PI's reference material and other related background material, develop multiple proposals for scope and approach, and review this with the PI sometimes in multiple iterations. And this is important. In our experience, the ultimate desired outcome often arises only after these kinds of back and forth deliberations, and that often diverges from what is in the original written proposal. To conclude Inception, we developed a detailed letter of collaboration containing all of this material, division of responsibilities between our team and the PI, and that's then signed off on. Okay, so I'm going to shift gears a little bit now and introduce a couple of the projects. There are 4 projects in the slides, but due to time, I'm just going to go over 2 of them here. The first one is underway is the humanization of optogenetic proteins. So optogenetic proteins are proteins genetically encoded to respond to light at different wavelengths, and they can be expressed in human cells to control and monitor cellular function. Optogenetic proteins have potential applications in many areas. In the case of our collaborators at MIT, they're looking to use the technique to develop optical neural control prosthetics and for therapies for peripheral nerve injuries. The journal Nature actually described optogenetics as a method of the year back in 2010, and Science described it as a method, one of the methods of the decade. The problem with optogenetic proteins is that the optogenetic proteins of interest are microbial in nature and derived from algae. So when they're introduced into the peripheral nervous system, they invoke a huge lethal immune response, and so the therapeutic applications are blocked. So the approach we've taken here is to leverage recent advances in deep learning models to attempt to computationally design novel, non-naturally occurring optogenetic proteins that are compatible with the human immune system. In a distributed computational pipeline, we first prompt a generative ML model with crystallized 3D Opson structure and use it to generate many candidate protein sequences. A second ML model then evaluates compatibility by predicting the binding behavior to major histocompatibility complex molecules. The most compatible proteins are then passed along to AlphaFold, which then predicts the 3D structure and measures its similarity to the known algae-derived Opson structures. Finally, the best candidates are passed into the laboratory where they're synthesized and evaluated for in vitro function compatibility. One interesting aspect of this technique is that any protein that is compatible and shows even partial function is potentially a success in the sense that it is a lead that can be further optimized in the lab, effectively lowering the threshold for computational design to be successful. This technique relies upon a common distributed pipeline framework based on the array tools, and we expect this to become a common infrastructure element and pattern that we use for many applications in the future. Another project, the second project here is OpenDNA. High-accuracy molecular dynamic simulations of DNA have many potential applications in biophysics research, drug discovery, and nanomaterials. The key to using these models successfully in applications is to make it easy to customize them and improve their accuracy in different use cases and settings. However, the existing techniques are plagued by very complex codes that do not adequately separate the models from the simulation algorithms. This makes the techniques opaque and difficult to adapt and extend. Furthermore, the existing models are parameterized in a crude trial and error fashion, and there's no systematic way to optimize parameters based on experimental data. So OpenDNA attempts to solve this problem by taking inspiration from the machine learning community in a couple of ways. First, it seeks to simplify model building by codifying it in a declarative, domain-specific language independent of the simulation algorithms. By doing this, users can easily change the geometry of a model, for example, or add or modify potential energy terms without requiring the expertise of understanding all of the estimation algorithms. This is imagined to be very similar to how machine learning models like artificial neural networks are specified in modern frameworks such like PyTorch, and then shared in collaboration sites like Kaggle and Hugging Face. The vision is to create and encourage a robust community of users and applications, just as the ML community has done successfully, by making it easy to specify, extend, customize, and share molecular models. The other inspiration from ML is to use modern automatic differentiation techniques, such as the JAX framework, to evaluate simulations numerically and estimate optimal model parameters from different experimental data. For example, DNA melting curves, which measures the degree to which double helix is associated due to temperature rises. This is not currently possible, yet would be a powerful direct application of numerical optimization in the manner of machine learning model training, but applied to learning the parameters of physical molecular models. So together, these two techniques, easy model specification and customization, and parameter learning, could be expected to fuel major breakthroughs in applications of molecular simulations in the coming years. Okay, and with that, I'll thank you for your kind attention and open it up if there are any questions. Yes.
Thank you for your talk. As a student in scientific domain, we often struggle with the exact problem: how do we develop software that's usable not only by our group but the community as a whole? Are there opportunities for students for like workshops within this group that we can at least get our feet wet with this idea before potentially embarking on a larger project?
I've been here since April and working on standing this up. We haven't set those programs up yet, but my expectation, and I think the expectation of my colleagues, is that we would intend to provide things like short, intensive workshops around best practices where that's of interest to different PIs and constituencies. So yes, I'd be interested to hear also what particular things you'd be looking for.
Thank you.
Yes, Gerard.
I've heard from other people that they're interested in figuring out whether some of their projects could be served by SSEC, how people at JHU could participate or could benefit from this. Can you say something about that?
Well, SSEC is currently executing projects according to the Schmidt Futures process selection. So, and that's going to continue, we think, at least for the first year. At some point, that is going to open up and evolve, I expect, but we don't know exactly when.
restrooms are straight back if you go that direction, past the coffee bar. The men's is on the right, the women's on the left. And I believe there's also restrooms at the one level down. And also, the IDES Annual Review is now available on the symposium web page, right? It contains detailed information about all the speakers today, and along with some other great information. So please do check that out. Thanks.
So thank you, and I think everyone for the really dramatic presentation. Next, I'm going to introduce Peter Lawson, our colleague here at this library for data and visualization, and he's going to give an update about Sheridan data services, but also about LoveDataWeek. We have had a lot of fun this past spring working together on an ChatGPT panel as part of LoveDataWeek, and I'm certain it will be another wonderful week here next February, and now the floor is yours, Peter.
Yeah, that's. Oh, wait, I thought something else. Question. How do I cast through the screen? I've got nothing. Oh. Yes. Sure. There we go. Okay. So hi, I'm Pete Lawson. I'm the Data Visualization Librarian over at Data Services in the Sheridan Libraries. I'm going to give an update and a little background on what Data Services is for those that aren't familiar, some updates from the Sheridan Libraries themselves, and talk a little bit about our upcoming event update. So to jump in first, I want to introduce Data Services. So we're a unit in the Sheridan Library supporting students, staff, and faculty along the entire research data life cycle. We've got a few modes of support which I'll go into. But primarily what we support is finding, using, managing, visualizing, and sharing data. We have a few mechanisms for doing that. With respect to finding data, we provide consultations and training. We also can receive and mediate data requests. In particular, in response to the New AI Institute, the library is taking a critical look at our catalogs and our data collections to better respond to and serve that community. So if there is data or collections or publications or journals that are not currently provided by the library that you're interested in, you should reach out to us. We can help as we sort of coordinate that response. We also help provide youth data use, and particularly data cleaning and analysis through some training options in Python and R. Data management, we facilitate development of data management plans. So we have something called the DMP tool, which allows you to submit a data management plan that we can then review and provide feedback for. We also support data visualization and data sharing through the research, Johns Hopkins Research Data Repository, which we maintain. I'll touch on all of these things in a little bit. So first off, our interactive webinars. So our modes of support are primarily two. We teach webinars in introductory skills around these 4 areas, and then we also provide one-on-one consultations. So in terms of our interactive webinars, we provide tutorials on programming. These are generally 2 to 4 hour, fairly intensive interactive webinars. Our programming is on data cleaning, data manipulation, data visualization, and these are exclusively in R and Python programming languages right now. But if there's any other languages or skills or of interest, we're always expanding our curriculum, and you can always reach out and ask. We also teach geographic information systems, GIS, and mapping. We manage the Esri site license for the institution too. So if you're doing any work with GIS, you have questions about GIS, you can reach out to us. Or if you're doing any geocoding and georeferencing, we help with that as well. We also teach reproducible research, primarily due to notebooks, our Markdown, GitHub. We're also expanding into Porto and more modern techniques like Gerard. And we also provide training and consultations around managing your code, your data, and your documentation to ensure it's reproducible. And then finally, we provide support for data management. This is, in response to the new NIH directive for data sharing. There's an increased need to have data management plans. We can help you with writing those data management plans, reviewing them, and then responding to any questions from program directors. If you get a response that says it needs to be changed in some way. We also help with human subject data. So if you're working on de-identifying human subject data for data sharing, that's something we can help consult and support. So in terms of our individual consultations, we provide consultations around all of these areas, just some highlights. Discovering and accessing data, again, we can help you understand what data is available to you through the library, what data is out there that is openly accessible, and then we can also mediate requests for data if we don't actually have it as an institution and you're unable to get it. We can also help with licensing. So if there's a data set that is at another institution or another government or entity, and you need to get those data use agreements in place, work with JHU Legal, we can facilitate that and get you the right places so that you get the data you need. We also, I'm the data librarian, I'm data and visualization librarian, so I teach a number of courses and provide consultations around designing and implementing data visualizations, best practices, creating accessible data visualizations, and I also teach a number of workshops around that in R and Python. And then finally, as I mentioned, depositing data, preparing data management plans. This is probably primarily our largest area of support, and so we maintain the Johns Hopkins Research Data Repository. So we can facilitate data sharing, but we can facilitate help with writing and reviewing those data management plans. So as I mentioned, we support data, and one thing we have is called the JH Libraries Data Grant. This is a small fund that facilitates data purchases for individual researchers. So if there's something that you're interested in and you don't have grant funding for it, it's not available at the institution, you can apply to data grants, and depending on availability of funds and your application, we'll be able to fund small purchases. So as I mentioned, we maintain the research data repository at Johns Hopkins. This is an open access research data repository specific to Johns Hopkins. It's open access. Deposits are free up to 1 terabyte. Greater than 1 terabyte, you just reach out to us and that cost can be negotiated. Each data collection gets a DOI, a persistent identifier. So if that is a publisher requirement, reach out to us and we'll be happy to facilitate that. that. And not only do we just take these deposits in, but we help with the curation process. So we'll ensure your documentation is complete. We'll make sure that you have the proper code books and data descriptions and get everything set up for preservation and sharing. So some updates on our in-person resources. So we are actually transitioned to a fully remote unit. So we do all our consultations and teaching over Zoom. However, we do maintain physical systems in the library. We were in the A level of the MCell library, but we're moving over to Brody Learning Commons on B level. And we've updated some refreshed workstations over there. So we've got 4 lab computers that will be available in the spring. These are 5820 workstations with Xeons in there, RTX 8500, so 24 gigabyte video RAM, and 128 gigabyte memory. So the idea here is that if you don't have the compute resources within your own lab or at home, it serves as sort of a bridge between working on your laptop and getting to HPC. So if you've got some research ideas, you need to just come and run something, and you're just memory limited on your laptop, but you're not ready to go to HPC, swing by our lab, and we'll have them properly set up for whatever machine learning or analysis techniques that you're shooting for. So as Ting-Long mentioned, we also are running our LoveDataWeek. This is an annual event. It's International LoveData Week, so celebrated across the world. Each academic institution hosts their own particular set of events. So Johns Hopkins will be hosting ours February 12th to 16th, 2024. And this will be sponsored by the JH Libraries, Institute for Data Intensive Engineering and Science, the Agora Institute, and the Alexander Grass Humanities Institute. So we're going to have a week-long series of talks and panels and workshops celebrating open data. So we'll have a lot of interesting panels about researcher experiences sharing data, the difficulties navigating patient identifying data, and we'll have some talks around data ethics as well. And we have some keynote addresses on us and our own Szalay, we're really excited to have. So some more general library updates. We are excited to welcome Megan Forbes as our new program manager for the Open Source Programs Office. So the OSPO is been operating, I would say, in name only for the past year or so. But we are actually the first university OSPO, Open Source Programs Office in the country, funded by Alfred P. Sloan Foundation. And you might know OSPOs from industry, but we're bringing it into the academic sector. So we're hoping to see a lot coming out of this office. Megan is doing a lot of work with stakeholders now to help develop our programming and support for open source. But as it stands right now, the OSPO provides tools and resources around the use of open source within the university, and they also maintain the university-wide GitHub Enterprise account. So if you're using a personal GitHub account, just be aware we do have an enterprise account. It does have additional functionality. So if you are interested, you can reach out to me, and I will get you set up on that institutional account. And then I just wanted to highlight a new database that we've recently subscribed to institutionally, Dimensions. Dimensions is a research analytics tool platform. It's essentially a super group of all academic publishers. So you might use something like Elsevier or these other publishers where you go in, in Science Direct and you find specific sets of publications. Well, what Dimensions has done is it aggregated across all of these different publishers and sets and indexed everything. So it has patents, it has publications, it has grants, and they're all integrated together. So you can actually use it to see how your research stacks up because it's all indexed. You can see, using network analysis tools, how you connect to other researchers, help find collaborators, find competitors in your space, and then help develop strategies for publications or maybe lab approaches. So it's a really nice tool. I highly recommend you go check it out, and you might find it's very useful. So So that's it, just some brief updates, but I will take any questions, and if you can't think of one now, then I'll direct your questions to dataservices@jhu.edu, and you can check us out at dataservices.jhu.edu. I'm sorry, I just put the web page, all right, not nice. dataservices.jhu.edu is our web page. Yeah, so I think we had one good example. This was related to data visualization, but we had a researcher come to us that had a publication that was not accepted. It was actually rejected because all of the data was in tables. It was public health data that was very, I would say it was very information intensive, and it would have benefited from a more impactful data visualization, kind of aggregated and summarized some of that information. And so through consultations with data services, they have been able to rework that manuscript and resubmit it, migrating those tables into some very, very impactful visualizations. So they're waiting to hear back, but we're optimistic that will get accepted. So.
Find their seats,
You have
please.
flyers to yeah. Which is also right. So we have good working agreements with the
There you go. That's what everybody's attention. So, the real reason it's so cold in here today is because we wanted you to know what it was like to be on the David Letterman show. All the young people have no clue what I just said.
I thought this was our new data set.
Right. Well, he was notorious for keeping his studio really cold. Anyway, so we're ready to get the next session started. I'm Anita Tara. And when we start, so we're going to start with the seed funding of the seed project. So IDES has an annual seed funding program that provides small 25K grants for pilot projects that hopefully become seeds for larger funding proposals. So that, I think the next cycle is going to be announced later in November. And they usually do by the end of January. So yeah, keep an eye out for that. So we're going to get an update on one of the seed funding projects. See, the first one is going to be not just about remote vital sign monitoring, but truly remote vital sign monitoring. And it's a project led by Chen, but presenting today will be Angelo. So, is Joseph here? Okay, please come on.
Okay, can you hear me in the back? Excellent. All right, let's see how this works. Hello everyone, it's an honor to be here, thank you for having me. I work at the Johns Hopkins Applied Physics Lab. Raise your hands if you've been there. Oh, yeah, we got something nice. Yeah, so we're over in Laurel, right outside of Columbia. And the PI of the grant is at Chen, who works at the School of Medicine, but a team of engineers and scientists at the Applied Physics Lab put together this proposal to work on expanding the capability and scalability of something like remote health monitoring. So the seedling is coming from actually a much larger project that was really had a really wide field of view, figuratively speaking, and we pitched it in 2019, saying, what happens if biosecurity threats, etc. 2020, a few months later, the pandemic came around, and we said, all right, switch this title slide. So, all right, stop the next one, because we're not ready, right? This was all an initial investigation to how workable and usable some of these solutions might be. And so, in the original pitch, it was this idea of like, there are all these avenues that could benefit from a non-contact, more robust and continuous way of monitoring health. In order to say, wait a second, don't get on that airplane, wait a second, this person is the doctor right away, wait a second, don't leave your house, you're coming down with something, etc. Some kind of monitoring, hopefully that, and also there's a biosecurity threat component here, where you are not strapping on, things that are thrown away, or things that you're strapping onto the next patient, etc. So this idea of non-contact continuous was very attractive to us. And for IDES in particular, we wanted to focus on just a part of this product view, which was this idea of like democratizing healthcare and giving access to all, because we all have a large portion of already have a really, really intelligent device on our hip, which is your cell phone. And so that was the idea. All right, really scale this down and see if we can make some action and progress. And so the general approach here, obviously in the clinic in some healthcare settings, is the conventional static approach where you get this really poorly sampled time, questionable robustness approach, or the continuous approach where you are highly connectorized in a hospital bed with professional equipment that generally takes professional staff to implement. And so the proposed approach in that original project was it's continuous, could be seen smart, I'll say, using computer vision to notice if you're coughing. It provides much information in a quantitative way to a healthcare provider or to yourself to make an informed decision. And then the big, hairy, audacious goal is to provide a wellness status through extracting these features into machine learning. Okay, and so we recruited many participants to a preclinical study with the help of Dr. Chen. The goal is to connect them to ground truth sensors here on the right. So if you're familiar with any of these, you'll know the joy and the headaches of using something like a BioPack system, the clinical grade line array, and commercial sensors. These are all contact. And then compare them to a suite of novel sensors that we saw in the literature were promising great results. We said we'll potentially test ourselves and compare them against each other and against the ground truth sensors. We have thermal camera, hyperspectral imaging. There's a suite of RGB cameras here with high definition, a commercial DSLR camera, think of like your photography camera, or your cell phone, both in a selfie view, so about a foot away, or rear facing about 10 feet away. This is all synchronized using PsychoPi and the LabJack to ask the participant to do certain tasks. Almost all the tasks are just breathing related so that the patient can stay focused and still, really. But there was one section where there was a cardio challenge where there's a stationary biking or pedaling. So there's some amount of motion, and there's some, again, challenge to the heart itself to raise the heart rate and get a change in signal. This is part, I'm going to go a little fast here. I realized I had too many slides last night, so we're going fast. The here in green is really what we're focusing on for this ceiling grant and what we're able to push through. You can't read any of this, I realize from right there, but the idea is to collect all of these sensors at a global timestamp, have them synchronized, have the data harmonized so that they can speak and be compared with each other and other sensors. This is a harder challenge than it sounds. Perform feature extraction, that's what I'm really going to focus on here today. And then eventually, when we have all these trusted quantifiable data sets, you can then perform machine learning, categorizing sickness, etc. The participants collected are node, our most predictable participant was a middle-aged white woman. We tried hard to recruit non-white participants. This is a challenge. You might be familiar with that challenge. But this was the spread. This was over 2 years. And I want to say that all the analyses I'll show you in just a second are subsets of this set, which, and in particular analysis will be a subset of that subset. And so the N overall is 91, and then depending on how you slice the data, again, if you're familiar with large data sets and the trouble in having consistent, fully complete data sets is quite difficult. Here's an example of a Canon DSLR. Again, we took the latest and greatest proposed method for using an RGB camera to pull out photoplethysmography data, so PPG, which is just like what your pulse oximeter does in the clinic, and it's click to your finger. We said, all right, we'll use that algorithm, do a little bit of tuning, how good can we get? And we saw a mean absolute error of about 8 beats per minute. And we said, well, that's okay, not great. Then we tried, again, using just subject matter expertise, knowing a little bit about the algorithm, what can we filter out? If we trained our computer vision to know that if someone's coughing a lot, simply don't trust that data. Or if their face is completely obscured, don't trust that data. By applying some of those filters wholesale, we're able to get it down to 7 beats per minute. Okay, that's fine. This is for a particular sensor, a particular scene. There has to be a way to test these thoroughly. And it turns out there is a way. There's a battery test you can do. There's a published repository called PyBHR. This produces, it has implemented the latest and greatest algorithms for a few of these methods. And we said, all right, if we take the method we chose, compare it with the best algorithms, parameter tuned, 198 parameters tuned on the right here. These are the parameters of interest. All of them beat, so this is for the cardio challenge task. So when they're performing cardio and their head is bobbing around, we get a mean absolute error of about 20 beats per minute with our hand-chosen method. But if we compare it to all the methods and pick the top 10, they win by about 10 beats per minute. And so clearly, there's something to be won by parameter tuning and choosing slightly different algorithms here. That's okay. If that's true, we again have this suite of sensors. What happens when we change sensors? And so in the blue, we took that DSLR optimized algorithm, we'll call it tuned parameters, used it on the iPhone selfie data. This would be the kind of setup we would do to take at-home, highly accessible vital sign monitoring. If we apply it to the iPhone, we're getting about a 30% error rate, versus if we just train the data, the algorithms on the iPhone itself, we're getting 10% or less. And so this is telling us, okay, scenes are very important. This is a sensor-based problem, but it's also the fact that this is now filling up your camera with pixels. You can't read any of this, but on the bottom are all the parameters that define the tuning. All of these down here. And what we basically learn is that for the Canon, there is a focused subset of parameters that seem to do very well. It's not a chaotic space. A slight tuning doesn't give you a dramatically different result. But as you can see, when you change sensor, all of a sudden we do get dramatically different results based on what tuning parameters, what algorithms you use. The next question was cardio. What happens if we train it? Motion is a classically difficult parameter to tune for here. If we train the algorithms on the cardio section and then apply it to the cardio section, we get the error in blue, which is around 15% error. And then if we train the algorithms on all time and all tasks and apply it to the cardio section, we do slightly worse, so the spread is a little better. And the key point here is that if you were looking to do any of this kind of computer vision processing, that motion remains a challenge at large, and that you would have to apply some kind of pre-processing, some kind of intelligent solution here. You could not simply tune your parameters to say, okay, take a slightly smaller patch size, take a slightly smaller window of processing. None of that seems to help us in this regard. Next up, so skin tone is a classic problem for measuring heart rate, for measuring SpO2, and some kind of imaging or color sense modality. So using an RGB camera, you can quantify color, obviously, and especially if you have a white balance camera. So the method here is an ITA, or individual topology angle. which basically just means where in color space does this color lie. And you can then break it down into skin classification. This is just like a dermatological Fitzpatrick scale, if you're familiar with that. And we had the question, okay, if this is generally a problem at large, would it be train these algorithms on a skin type and then apply it to that skin type? And so the left is the tan group, and in blue is if you train the algorithm for the tan group and then apply it to the tan group. And so you can see compared to the red, which is just this general algorithm, it performs slightly better. I want to point out that this is an N of like 5, and so they're again a subset of a subset of a subset to have this completely comparable really shrunk down our sample size here. So that's frustrating, but you can see that the statistical difference is within the noise of having such a low N, and so ideally we get more participants and continue this kind of work, but you do see some kind of trend skewing towards a lower error. I think the key takeaway here is really that this is something that is quantifiable in the scene. So I mean, your pulse oximeter probe that's clipped to your finger has no idea about what skin tone you have, and so it cannot act on this, but something like this could. I'm not going to go into blood oxygenation theory or anything like that, but I would basically provide 2 key results, which is an RGB camera performs just about as good as your contact biopack sensor, if you're familiar with that. This is somewhat cheating because we trained it on the ruler itself. We trained it on the clinical Acutor sensor. And so, yes, with a little bit of leg up, you can certainly beat it. But more impressively, there are algorithms and things we've implemented that provide a calibration in scene, which is just a color checker chart to provide some kind of global correction for the light, for the sensor itself. And then again, we're able to get mean error around 0 to mean absolute error a little below 4%. There is some skin dependence here. Now I'll just quickly say that the clinical grade sensors themselves are classically faulty for different skin tones. And so without doing a blood gas test to verify these results, it's just a difficult problem to solve. I said we had a suite of sensors, and this is the suite of outputs that we've been generating, measuring it from several yards away with laser vibrometry, doing machine learning algorithms and thermography. If any of this is of interest, please reach out and please contact me. I'll quickly say that the future work is really going to focus on this guided processing. So using more intelligent information from the scene itself to apply those tuned parameters or to choose up your algorithm in general, as opposed to thinking that a single setting can solve all your problems. Thank you very much for your time. I'm happy to answer any questions you have.
How about data from smartwatches, which are contact but kind of not medical grade? Have you thought of including those?
The short answer is no. It's actually harder than you think to work with something like Apple data or Garmin Watch. I don't know if you've done that.
I'm sure it's impossible to work with.
Yeah, they're not super willing to say, here's the raw data and all the sauce that goes with it. They don't like to do that, unless you pay them, which I already know. The short answer is no. We haven't done it for something like a Fitbit or something, but we have done it for the things you can buy at CVS. We have done it for those. And so for heart rate, it tends to do a little better. God forbid that finger moves at all, because then your data goes to nothing. It's also not very good at signaling me that is bad data. And so if I shake my finger at 100 beats per minute, that's going to be the heart rate that it reads. Right? It's kind of silly, but it makes a lot of sense though. There's a lot of caveats that come with using this kind of data, which is, if you go to the clinic and do your blood pressure taken, they say, both feet on the floor, stay absolutely still, don't breathe like a wild person. They'll give you guidelines to follow. In a take-home setting, those are a little harder to implement. And so having, again, some kind of scene, intelligent modality, I think is the way to go. Okay, thank you very much.
So next up, we have an update on ARCH, Advanced Research Computing at Hopkins, which is a data-driven, high-performance facility that is managed under the auspices of IDES. And Jaime, Combariza, the director of ARCH, will be giving the update. Just a quick announcement. If you park in the San Martin garage, please see Elliot back there to get the validation or whatever. So.
Advanced Research Computing Center, formerly MARCI. Actually, the facility is still called MARCI, but the group is called ADGE now. And this is an update on what we did on FY23. This is our 9th year of operation going into 10. And I think that year after year we become more powerful. And that means computational power, not anything else. Okay. So I keep this slide up because it basically summarizes what we do. Part of what we do is not all of that. So we provide all kinds of cycles, CPU, GPU, large memory, and storage. We have like 15 petabytes in storage. All of that for parallel storage. We provide containers that are very common in the last few years. And also, faculty and users have requested a lot of interactive access to the compute knowledge because people like to interact the application with the data, manipulate the simulation for the experiments, and then decide what to do. So that's also very good. It's probably some 20 or 30% of the work that we do these days. We use Open on Demand for now. That's a very powerful application funded by NSF. We provide fast data transfer from like NCP or any other facility to MARCI, 100 gigabit per second, dual connectivity. We have more than 100, maybe 200 or 300 applications already installed, ready to use. So that is going to save time for most of the users. We have a co-location center, mainly the Whiting School of Engineering is using that. They have actually Whiting and public health. And then we provide a lot of scientific support. It's not only the hardware, it's also the support for the effective utilization, not only of the resources, but also of the applications. Okay, this is not too bad. So these are the resources as of July of 2023. So every year we increase 10, 15, 20%. We have more than almost 42,000 cores today, which will give us a very powerful system with about 2.17 petaflops, and that will probably put us next week, a couple of weeks from now, into the top 500 and 400 or so place. So that's very good. We provide storage from different systems and so forth, so all of that is probably known to all of you. Some of the metrics. So we already have more than 400 faculty members of research group at Hopkins using the facility. Actually, no, in the facility. Maybe not close to 400 active. So basically in a month, they will connect and they use some of the resources. We have more than 1,000 active users. Again, they connect to the machine and use some kind of CPU time. And the utilization is normally about 90%. If you check today, it's 100%. Usually during the week, it's 100% fully utilized. And during the weekend, it's a little bit less than that. A lot of publications that have been produced using ADES, Rockfish in this case. Last year, we hosted about 20 classes and workshops, so it's also increasing. And there is a rumor in there that MARCI or ADES is Rockfish is very unreliable. I don't know where that comes from. We checked and in FY23 the uptime was 97%. Yes, there was a week or two where we had to do some maintenance and the system was down, but still it's 97% uptime. With College Clancy, we have been talking about going through all the departments to tell them about Rockfish. Quite frankly, I'm hesitant to do that because, as I said, we have over 400 faculty members already using the facility, and the system is at 100%. So if we are going to add more people, we need more resources, and at least for Rockfish, we're full. Utilization, this is the same trend year after year. Last year, Whiting was the winner by almost twice as much as Krieger. The public health is decreasing the utilization. They have their own facility, so most of these faculty prefer to use that facility. But utilization is increasing year after year. As as I mentioned in the previous slide, about 240 million hours last year. The model. Again, this is a repeated slide, but we like to use this because we have three main groups contributing to Rockfish. The first one on the base of the system was an MRI grant. Later on, we added a DOD and the big grant, and that produced some nodes. And then we have the deans of the four schools, Whiting, Regan, School of Medicine, and Public Health. They provided some resources for all their faculty. And then we have the small grants and new faculty lines. So we have over 400 nodes on the first group, over 100 on the second group, and the small grants and faculty lines about 298. But if we take the grants, which are faculty based, and the new lines, so 1 and 3, for one, the schools contributed about 20%. So if we take those two and we add them together, I can borrow this statement from another university, and this is truly a core facility that is from faculty for faculty. This is open to anyone that is a faculty member at Hopkins. The condo model. I have a lot of questions about this. So basically what we want to add is we have a base system and we want to increase the computational power by adding more hardware. And new faculty members or faculty members that have grants, they can add any compute nodes, CPUs or GPU or large memory nodes. And then we'll increase the capacity for everyone, not just for the group. The group is going to get a second allocation, or if they prefer, an increased allocation. For instance, Charles has like 3 allocations from different grants, so they can get a lot more compute power. So that's it, the basis of the condo model, and we have about 50 or more faculty members already using this condo model. So it's working beyond my predictions because 3 years ago I thought that if we build a system with about 900 nodes, that will be difficult to fill. Today, yearly, we run out of space. We need to start building a new cluster. I mentioned interactive computing, so we use Audio on Demand. That is an application that you can use in your browser to connect to a compute node. And many people run the simulation in batch mode, and then use Audio on Demand to run like a Jupyter notebook or any application to look at the results. And then they can decide if I'm producing the result that I want, or it's not, some of the parameters are wrong, so I can kill the job, or I can continue the job. And it is also used very good for people that have graphical applications. If somebody wants to use Abacus or MATLAB, they can use this application, connect directly to the compute node, and run their application. Again, the GUIs will run really fast using Audio on Demand. So again, this application is heavily used these days to do interactive computing. This is an example using MATLAB. You can run some code and produce beautiful pictures that you can save into a file, and you can do that with many other applications that I showed you. And this was mentioned before. We are also involved with several groups doing some reproducible research. So what we do is we create a science gateway, basically a portal, where people can upload the data, and they can share the data with whoever they want. Most people want to keep control of who is going to access the data. They can create collaborations. They can use that for validation. And we have a good example in a faculty member in civil engineering. They had an audit from DOD. So this person wanted to run their codes in the portal, but also wanted a container because he wanted that to be portable. And that's the portability of this reproducible research. We're working with about 10 other universities to create a nationwide community of practice. And education, as mentioned before, we had about 20 classes last semester. This year we have a new small cluster dedicated for teaching. So people don't have to interact with the production system because if it is completely a class that is in biomedical engineering. They wanted to use GPUs. So we took two of the GPUs that we had, produced a so-called multi-instant GPUs. That's 56 GPUs per node. And then the class with about 100 students is making use of this. The future, as I mentioned before, we need to start planning for the new cluster. So we worked on a new RFP, and we want to include, again, we have a plan for sustainability, so the BIS are going to provide some money. And then we are already applying for grants. Next year we're going to apply for another MRI. And we're going to use small grants, and again, new faculty lines are already planned for next year. They will be part of this new system. So in short, I think that we have provided some good examples for AI and some support, scientific support, and the facility, the infrastructure to produce that. Thank you. If you have any questions, I'll be happy to answer, or I'm going to be around.
So apparently somebody from facilities is on the way to address the cold freezing situation. So IDES also runs a summer student fellowship program, and next up is a presentation from one of the summer student fellowships. Unfortunately, today, usually this symposium is on reading day, when the students are free to join us, but today unfortunately they have classes. So Sampath, Rapuri, who is the person who was a summer student fellow, could not be here. So he has sent a video instead. Get him wrong.
Hi there, my name is Sampath Rapuri, and this summer I was fortunate enough to be chosen as one of the 2023 recipients of the IDIES Summer Student Fellowship Prize. With my mentors Gerard and Dr. Stevens, I was able to learn a lot more about my interest in data science and apply it to an incredibly relevant topic for patients across the world in intensive care units, that is pulmonary embolism. Pulmonary embolism is a life-threatening condition that results from the embolization of deep vein thrombosis in the lower extremities, and which moves and migrates into the pulmonary arteries. And so that basically means is that a blood clot forms in the lower extremities, travels to the pulmonary arteries, and obstructs blood flow there. The alarming truth about pulmonary embolism is that 15% of patients in ICU units around the world can actually contract this condition, and it is a leading cause of death in ICU patients. What's more is that the clinical signs and symptoms of pulmonary embolism, also known as PE, lack sensitivity and specificity, which mean that they do not help in discerning pulmonary embolism from other conditions. To confirm pulmonary embolism then, physicians often subject patients to numerous and often specialized tests, which add to the complexity of this already hard to diagnose condition. What compounds this even further is the inadequacy of current risk assessment methods to see which patients are at risk for developing pulmonary embolism. Physicians are then faced with a critical need for better and more accurate predictive tools to foresee the onset of PE. And that's where my project this summer fell into. I was trying to develop and validate a computational model for the prediction of pulmonary embolism in critically ill patients. To begin with, I employed the EICU Collaborative Research Database, which is a large multi-center database with demographic and physiological data for over 200,000 patients collected from 208 hospitals across the United States. But we employed this particular data set because of its large patient size and high granularity time series data. From this data, using a keyword search in the diagnosis string entered by the physician for each patient, we looked for the keyword pulmonary embol to identify all of our pulmonary embolism patients. We cross-checked all the patients by their ICD diagnosis code and made sure that the number of patients we got was equal to a search purely through the ICD diagnosis code for pulmonary embolism. From this, we gathered and analyzed a total of 2,799 unique ICU stays where a diagnosis of PE was determined. From these patients, due to a lack of reliable determination of when the PE truly took place, as the physician can enter in days or even hours of days after it first truly occurred, we stratified our time-dependent data into 3 different observation windows, if you will. So 12 hours, 24 hours, and 48 hours after initial ICU admission for our patients. In total, our aggregated data contains laboratory data, demographics, vital signs, comorbidities, medications, and more. And for lab and vital sign data, we took care to make sure our aggregated data all had the same units to ensure compatibility. Furthermore, for these physiological variables, with the help of Dr. Stevens, I made sure to look through all these different distributions to make sure that they fall within a plausible range. Additionally, for our time-dependent data, we chose to create simple summary statistics to capture the data over each observation window. So we looked at the first and last values in the time period, the mean, the minimum, the maximum, and the standard deviation throughout each time period. We also explored more complex features such as Fourier transform coefficients from these highly granular time series data, some of which occurred every 5 minutes or reported every 5 minutes. However, after testing them out through the modeling process, we saw no significant increase in model performance, and as a result, for the sake of model simplicity, we decided to omit these features and offer the simpler summary statistics for hopes of better interpretability at the end. So after pre-processing, which included simple mean and median imputation for numerical and categorical features respectively, we normalized and scaled their data. Using these data, we then trained multiple different machine learning algorithms, which included decision trees, gradient boosting models, which included XGBoost, CAT-boost, gradient boosting, and more. Random forest generalized linear models like logistic regression, support vector machine classifiers, and artificial neural networks. We then compared these results to the current gold standard risk assessment methods that physicians use, which include the modified Wells scoring system, the revised Geneva scoring system, and the pulmonary embolism rule-out criteria. All 3 of these models allow the clinicians to estimate risk based on a discrete number of predictive factors that are used as inputs in multivariable logistic regression equations. However, all of these models have limited predictive accuracy, in part because they may not consider the physiological complexity of acute renal patients, and that's one of the challenges we attempted to overcome by surveying a large variety of models. One issue, though, was when we implemented these risk scoring mechanisms as a kind of control to compare our modeling results against, was that some of the features that the models used were not available in the data, and so we had to modify the risk scoring mechanisms according to the limitations of the data. Interestingly, speaking to the results, logistic regression actually proved to be our best performing model by AURC. That's reported in figure 1, you can see. We expected that other models actually, like some of the gradient boosting models actually, would be able to capture nonlinear relationships that exist in the data, and thus be better suited for modeling, as logistic regression simply cannot do that mathematically. However, this was not the case. In figure 2, we also explored feature importance. For other models, other nonlinear models, we explored feature importance using the Shapley additive explanation values to look at the clinical importance for each variable. However, for logistic regression, as it was a linear model, we simply observed the absolute value of the coefficients to establish feature importance. And looking at these top features, we saw multiple that coincided with the features that were used in the above risk scoring models. We also saw some that were to be expected, like respiratory rate, oxygen saturation, and the presence of certain comorbidities. We also saw certain features that were surprising, some due to their rank and some due to their novelty. For example, we did not expect the presence or the kind of when the patient takes a type of antibiotic called quinolones to rank so high. As you can see, it's the number 1 most important feature. Additionally, was the importance of calcium values. Both of these are reported in the literature, but not with this degree of importance. We hope to further explore these relationships between these features and provide new and insightful insights for clinicians to use in the management of pulmonary embolism. Right now, I'm currently exploring the Precision Medicine Analytics Platform dataset, a large EHR dataset co-developed by Hopkins as a source for external validation to see if the models we develop through this EICU dataset generate as well to new patient populations and still proves useful as a tool for clinicians. Overall, speaking of this project as a whole, I do want to say that it was an amazing opportunity to learn a lot about overall project design from the stages of hypothesis crafting to testing and modeling. And now, as I stand here by video, I begin to complete the cycle that I initially went through months ago with Dr. Stevens and Kirby by sharing and presenting some of the results that I have through this summer project. Some of the challenges that I was facing through this project, though, were dealing with the messy, unorganized nature of EHR data and a lack of consistent standardization. Kind of contrary to what we usually deal in class of students, these data are very messy, complex, and full of noise. And trying to wrangle with it takes a while, which I didn't realize at first initially. And so that was a much needed experience for me to see kind of how real world data looks like and how to deal with it. One more thing that I had to deal with was working with large amounts of data as was relevant in this project with an additional constraint on computing resources. This taught me to become kind of learn to deal with those restrictions and furthermore become efficient with my code and the resources that I do have. And that kind of would help me design my code throughout this entire process. Lastly, guiding me through all my questions and concerns were my mentors, Kirby and Dr. Stevens, to whom I owe a lot to, and this project couldn't be done without them. Well, thank you so much.
So our second summer student fellowship presentation is by Kim. It's about intelligent microscopy methods using electron energy loss spectroscopy. Could the poster presenters please start lining up? That's nice.
Hello, my name is Kim, and I was another one of the student summer fellows, and I'm really excited to be presenting to you today my work on intelligent microscopy methods for characterizing intermediate states using IDES. So to give you a little bit of rationale behind this experiment, in the world of material science, there's a very powerful analytical technique called transmission electron microscopy, or TEM. How TEMs function, in essence, is that it transmits a thin beam of electrons through a sample, and depending on how the sample interacts with those electrons, we're able to obtain information regarding the structure and properties of the said material. Another very popular technique used in conjunction with TEMs is electron energy loss spectroscopy, or EELS. And EELS takes into consideration the inelastic scattering of electrons, and from that we're able to learn more about the electronic properties of materials. So this is an example of some noisy EELS spectra that we'll use later on in this presentation. But all of this is to say, in recent years there have been a couple of limitations regarding those two methods. Firstly, the rapid development of direct detection systems, as well as just the inherent challenging nature of the EELS technique. has led researchers to turn to machine learning in order to bridge some of these gaps in knowledge. The ML algorithm that we chose to focus on in this study was the autoencoder. And as you can see in this figure here, there are three main parts: the encoder, the latent space, and the decoder. What we really want to focus on is the latent space because it serves as a compressed representation of our initial inputted data, and it's also very useful for feature extraction. So with our autoencoder, we began by testing it using the Modified National Institute for Standards and Technology's DIGITS database. Very long name, but in essence what that is a set of around 60,000 training images and 10,000 testing images of handwritten digits from 0 to 9. To give you an example of what I'm talking about, these are 3 images from that set. As you can see, it's 5, 3, and 4. We added a little bit of noise to it, and after passing it through the autoencoder, it was able to reconstruct it with a much lower signal-to-noise ratio. An inherent issue that exists with traditional autoencoders, however, is that occasionally an error will appear where there are points in the latent space that don't necessarily directly correlate to anything in the inputted data. In order to address that, we turn to the variational autoencoder, which is very similar to the traditional autoencoder, except it applies a mathematical constraint to the initial data set, and it also is able to address these regulation issues by forcing the distribution to be a normal one. So for the actual experiment, we used a strontium ferrite perovskite crystal structure data set, moving from the MNIST, and this is an example of the latent space of that data set. In this algorithm, we used unsupervised clustering, which is why we didn't characterize those 3 states, but I would like to point out our model's success in being able to draw out well-defined boundaries for each of these 3 regions. This is some spectra comparison, similar to the first slide, between experimental data and simulated data, with the experimental spectra in blue and the simulated spectra in orange. I'll talk a little bit about how we got to the simulated spectra later on in the presentation, but what I really want to highlight with this figure here is the alignment of the peaks between the experimental and simulated spectra. Although there is obviously a bit of an offset, which could be due to a background subtraction error, the fact that the peaks align so closely is a strong indicator that these two spectra share the same structures, which is the initial goal of this experiment. So touching back on the simulated spectra once again, how we got it is we utilized the Open Quantum Materials database, which contains highly theoretical and idealized information on a wide variety of materials, including various structures of the SFO used in the study. And with that information, we plugged it into the finite difference method near edge structure program, which uses density functional theory, a computational physics-based model, in order to produce said simulated spectra. From this, though, we learned that because of the fact that OQMD contains such highly theoretical information, it's not necessarily the best in representing actual experimental data, which has obviously a lot of variability. And so going forwards with this project, we would like to compute the density functional theory calculations from scratch in order to better emulate this experimental data. However, this isn't to say that the OQMD work was useless, because it provided a very prominent first step in being able to create a basis for classification for these structures, as well as gave us the ability to take off-the-shelf information and produce spectra from it. And so I'd like to thank my mentor, John Hollenbach, as well as my professor, Dr. Takiri, for supporting me throughout this project. And yeah, I'd be happy to take any questions now, but otherwise, thank you so much for listening.
Thanks again. So the last thing before lunch is a very sane, orderly set of 2-minute poster presentations. Of course not, it's poster madness. I think we have a handful of people who want to present. Is there an order?
And this is the question I'm just reading to everybody. your poster title, that is when your 2 minutes will start, and you will hear my alarm go off when your 2 minutes is up. So, and the poster gallery will take place during lunch, which is after this, and the posters will be visible in the great hall over there, so we will be able to see them in person. Without further ado, here is our first poster entry.
Hi, I'm Sheridan. I'm postdoc in mechanical engineering, and I don't fully remember my title, but I think it's conditional analysis of energy cascade in turbines using a novel database. Okay, I think that's it. So in the past century, we've been trying to learn and study the future and physics of turbulence. So turbulence is what we see every day. In our life, like the wick behind the car, behind the plane, and also this very complicated patterns we saw in cloud and ocean. So this is sort of turbulence. Until today, we cannot predict it yet. So that is why turbulence is one of the remaining open questions for classical physics. Fortunately now, thanks to the computer science now, we are able to simulate turbulence with a supercomputer, and we're able to store this data using some big data tools. So in my research group, we using this data and did a lot of interesting statistical analysis. So if you're interested in this very classic but mystery physics problem, is welcome to my poster. And, thank you very much.
Hi, my name is Tom Hain, and my poster is called the SciServer project, and I can't remember the rest of the title. I'm a professor in natural sciences, and I study ocean currents. So ocean circulation is important for many reasons, but principal among those is its importance for climate. And over the last several decades, the information technology revolution has transformed ocean circulation and understanding of the ocean currents. For example, there's been exponential growth in the number of observations of the ocean. It's also transformed the way in which we can simulate and model the ocean currents, and there's been exponential growth in the resolution, the fidelity, the accuracy of ocean circulation models. But the two rates of exponential growth aren't the same. The ocean models have been growing 5 times faster than the observations. In fact, they're now essentially unconstrained by the observations. Despite that, the ocean circulation models have become harder and harder to access and use. And so our project, the SciServer project, is to build a database on SciServer that gives wide community access to this ocean circulation model. The poster is in the corner, you're very welcome, and it's my track.
Hi everyone, I'm Raj, I'm from the Cognitive Science Department. My poster is titled something like the high-dimensional structure of visual cortex representations. So what does this mean? So I study the human brain, I study how we perceive things, visual input. So one leading theory of visual perception is that our brain takes in a high-dimensional visual image, so like millions of pixels, and then over several stages of processing, it compresses that image and extracts the most relevant features. So you take some high-dimensional input and keep compressing it, and you extract a low-dimensional latent representation that it then operates with. But then, so we developed this technique based on PCA. So it's something where you can extract a spectrum of reliable variance in brain data. And with a large-scale fMRI data set that's coming out, we can basically take pictures of your brain in response to many different images. So I show you an image, and I record your brain responses. I show you another image, I record more brain responses. And then I can take these brain responses and look at this physical structure. So I can look at what kind of covariance spectra they have. And using this, at different stages of visual processing in the brain, we found out that the brain represents visual input as a high-dimensional infrastructure characterized by power law covariance spectra. So at different stages of processing, all the way from the input to several stages deep inside the brain, we see that the brain represents visual information with this high-dimensional latent code.
So instead of compressing visual input into a small set of useful dimensions, the brain actually maintains a very high-dimensional, expressive basic set of features that you can use to do a variety of tasks. And it turns out this sort of power law spectra has been found in machine learning as well. So neural network models with human vision, turns out those models which have higher dimensional data structure also perform better on many engineering tasks. So if you're interested in learning more, come to my poster.
Hello everybody, my name is Shu Hongwei. I'm a graduate student of the Applied Mathematics and Statistics department, and our poster is named Computationally Saving Methods for Approximating Error Bounds of Extended Common Filter. And usually for nonlinear systems, we know that the common filter iterates or preserve a pseudo-error covariance matrix, but that error covariance matrix will perform not really well if the system is highly nonlinear. And we want to get ways to approximate the confidence region of the filter estimating error by only one EKF runs. And our baseline method is the multiple path EKF run error simulations. And we found that when we can prove the system being agortic, it is possible to use just one EKF runs to have good estimation of the state estimation error distribution. And if you are interested in our poster, please visit us during the lunch break. Thank you.
Hi everyone, my name is Shorty Shao, and right now I'm a postdoc at Oses and under the supervision of Charles Manuel and also Denise. And my poster is about Johns Hopkins wind farm flow database that are targeted for publicly and easily accessible. Yeah. So right now we're in the era of energy crisis, especially after the conflict between Ukraine and Russia. And so wind energy is one of the resources that were added to this diversity in terms of renewable energy. And for numerical experts like me, so we typically use numerical simulations and to provide the database to data and for the people can evaluate for sex. Okay. So, but technically, we use high fidelity numerical simulation, like the large eddy simulation, combined with the actual line model to simulate the wind farm. Okay, the traditional challenge to this because typically the data is so large and it's not hard for the public to get access. So the aim of this database is try to make it publicly and easily accessible with the well-constructed web service so that people can access it easily. Okay. So currently we have two setups. So one is the wind farm in the neutrally bound layer, and with like 60 wind turbine there. Another is one with smaller amount of wind turbines, but in representative diameter cycles. So I think these are very useful to wind energy implementations. So if you are more interested on the details, please come to the gallery for more information. Thanks a lot.
Hi everyone, my name is Zan. I'm a second-year PhD student in the Applied Math and Statistics department. My poster is titled Random Convolutional Features and Hash-Based Learning for Multitask Image Classification. So what does all that mean? So we know that neural networks are a class of functions, and specifically shallow neural networks are the composition of a nonlinear activation function with a linear function inside of it. And the universal approximation theorem tells us that there exists, a set of parameters for these nonlinear and linear functions, or weights and parameters, that can get you arbitrarily close to any continuous function. So what we study here is what happens when you take the parameters for the nonlinear function and randomize them and fix them. Is there a combination of scalar weights that still allows you to universally approximate any continuous function? So the answer is yes. So in the context of
function fitting and data fitting, you can get arbitrarily close with a single hidden layer neural network of arbitrary width. If you randomly fix the parameters for your nonlinear function, you can just optimize over the scalar weights of your linear function and get arbitrarily close. And the interesting thing is that the order of convergence is the same for just optimizing over the scalar weights as it is for optimizing over the nonlinear parameters and scalar weights. And so we applied this theory to the image classification task in convolutional neural networks, and specifically we sampled the random patches, our filters as random patches from the data set themselves. And so instead of a neural network where you're optimizing your filters and the weights of your filters, you can just randomly choose and fix those filters, and then you save a lot of computational expense that you would originally have by doing gradient descent and backpropagation for your filters. And so we tested this on the IDES and the CFAR data set with analogous architectures of neural networks, so one convolutional layer and one fully connected layer. In one case, we randomly sampled the filters from the data set and fixed them, and then only optimized over the weights. And in the other case, we just treated it like a traditional CNN where we optimized the convolutional filters and the weights. And it turned out that randomly picking them was much faster and better in terms of accuracy. And then the benefit of this is the multitask classification. Since your random projection is unsupervised, you don't need to retrain for different tasks.
All right, it's 2 o'clock. We should begin. This is what we get. The man who needs no introduction, Gerard Lemson.
Hi, my name is Gerard. I'm the Director of Science for IDES and Social Director at SSEC. And I'll give the yearly overview of what happened in SciServer last year. So a bit of a background for people who don't know what SciServer is yet and why we built this. So, a good friend of Alex and Ray, he called it the 4th paradigm data-intensive science. Basically, these days we use large amounts of data from all kinds of different sources, and we use sophisticated AI, machine learning, etc. for the analysis. And we do this generally with lots of people, in large collaborations. So this presents some issues for science. Data grows exponentially, and you need people who really know how to deal with large data sets, both for storage, download, for analysis, etc. The data become public, so you need to do something with that. So people would like to combine large data sets. How do you do that? How do you get these heterogeneous data sets together? You need to standardize, etc. And the science is increasingly collaboratively. You want people to be able, from different disciplines, to participate in your project. You want to do it easily. So it was in the old days, where there was no centralized data stores, etc. People have their own laptops or desktops, and if you want to collaborate, you have to send data around. If you want to get expertise from somebody on databases, on AI, on GIS, on Python coding or other kind of languages, you would need to get them somehow to talk to each other. This is very complex. And people, you might go to n squared of kind of connections that you need to do. So a nice solution is something that we now coin a science platform, where all the data is in one place, computers in one place, people can log in, people can share data, people can protect the data from other people. So now you can work with your collaboration, distributed all over the world potentially, to get all their expertise together and work with them. This is what SciServer is. It's an example of such a science platform. For people who don't know SciServer, go to www.sciserver.org. You can register there. It's open to the world. What you then get is, behind this dashboard, you get file storage, personal, also shared institutions or collaborations that we know, or sometimes that pay us. We give much larger storage For you personally, you get 10 gigabytes at the moment. We hope to go higher of your own space that will be backed up. It's persistent. You get access to temporary sketch storage, terabytes, in a few years, petabytes. Not just for yourself, of course, but also you can collaborate easily. You can create a group. You can invite other users. You can share workspaces that you create with those groups, with those users. This is how we also give access to collaborations. They define the group. We assign storage space up to terabytes, tens of terabytes, some cases hundreds of terabytes if they buy the hardware themselves. And they can decide who can actually see the data and with what permissions. This all started 75 years ago, roughly, with the Strong Digital Sky survey, where the group here revolutionized astronomy in some sense by allowing people to write custom SQL queries against databases that were state-of-the-art data. SciServer goes beyond that, and that it also allows you to write Python, R, Java, C, Fortran, whatever you want to run, on environments that you can start up for yourself, server-side. Most people will use using Jupyter Notebooks, generally using Python, but you can run other languages underneath that as well. So that's basically a very fast overview of SciServer. If you're interested in using it, there's a bunch of user scenarios that we generally see. One of them is what was the original, big data dissemination. If you have a project and you have terabytes, petabytes of data, and you make us part of your collaboration, you may buy hardware in our data center, we can help you with turbulence. Lucas, for example, that has been going on since I think 2006, roughly. We're still working on that. There's a new one that I'll talk a bit later. They said it came online just recently. The relational database platform, we still expand that out as well. It's not only astronomy anymore. We have integration between relational databases, file storage. The whole goal of SciServer is if you don't have to download data, you can bring your code to the data using these compute notebooks. And there's CPUs and also GPUs that we don't yet hand out freely, but contact us if you're interested. We have DAS for parallel access. Some people here are experts on that. We use that Spark cluster. We don't have it at the moment, but we can easily spin it up again. So another thing is, within your collaboration, if you want to share resources, a project of that, an example of that is AMS, I'll talk about that. It's a perfect platform to bring data together. You just write the code, you grab the data, you store it locally, you can write code to manipulate it, to put it in common schemas, whatever it is what you want. I'll talk more about this later, but there are already external instances of SciServer as well. There was just one in our data center in Homewood 156, but the Precision Medicine Analytics platform, the School of Medicine has one that is HIPAA-safe. Planck Institute for Extraterrestrial Physics in Munich has one. So there, Rosita, X-ray satellite data. There is one at IDES behind their firewall. On the International Lab. Started more than a year ago. We have people who know how to build custom IDIES, very fancy ones. Poseidon is one of them. I don't know if it's actually on the poster that Tom has there. Other dashboards that we've made. And then we support education. It's quite easy to set up a kind of a workspace for a class, for a workshop. And so the practical genomics is going to run, I think, next week for a couple of weeks, and then using SciServer. Okay, what did we do this in the past year? So we had a new hire, Chen, who was already working with Alex before I joined, but he joined us now as at the IDES RSE, similar to the ones that Edward talked about. These are his expertise, and this is what he's also going to work on. We have moved SciServer, the web applications, to Kubernetes. I think Eric calls this your own option. We told other people if you want to have SciServer running locally, make sure you have Kubernetes, and then the installation is simple. It is indeed relatively simple. I was able to do it with Eric's help at some point. But this makes maintenance much easier. It makes interaction and demarcation of tasks and duties between our IT group and our application group segment. And so we have SciServer Kubernetes, but we have a larger web cluster with 5 nodes, I think, at the moment, maybe grow to 7. We are planning all our web applications that they will be ported to Kubernetes as well. The data center is going through updates. This is all ideas of Alex. We had 90 so-called data scope nodes that are now all being turned into 7 nodes with, I think, every 10 nodes have about 5 petabytes of data. Is that right? I don't remember. So we'll have 50 or 60 petabytes not long from now. We bought a few new database servers that replaced old ones that we have that are all now NVMEs. This is mainly for databases of people who want to have a database with us, so collaborations particularly. We start building a new compute cluster that is all running Kubernetes, not just for SciServer, but also pipelines that other projects want to use. We'll have 25 quad nodes, each of these nodes inside the quad node 128 cores and a terabyte of RAM. So it's going to be a large system. A first node of that is already available for benchmarking that we're currently doing, as a new compute domain as we call it. And as it was the SSEC that Edward was talking about this morning, just bought 4 GPU servers, each will have 4 A100 GPUs. They're intended for use by SSEC, but because they benefit from being in our data center, we'll make it available somehow through SciServer as well, with more restricted access. Okay, some projects. This is the kind of our diagram that we used in the past for showing all the types of science projects that we've been supporting. This is a new version of it that we needed for a proposal that I'll talk about. Here's just a summary of some of the projects that we're working on. The Turbines and Poseidon, they are the big projects with up to petabytes of data. They wrote us in their proposals. So that's, we get people like Mike working almost all the time for a Turbines project. There are some smaller ones, there's the National Science Data Fabric, there's HCM Deck from David, democratizing data that Julia and Lenny will talk about later. We have extended our NIST collaboration in the PrEP framework. There's some nice project there where we do some AI and data in their disaster and recovery investigations. We're participating in some of the SSEC projects. Since we have RSEs already for many years, there are certain projects that suite features proposes to SSEC, and we have people available, say 50% of the time, like some of the people like Lemson or Ryan that we know in the past. They can come and start working on those projects. There's a few other ones, smaller ones, where we have Albert heard about Oida this morning. Some proof of concept projects are going on that lead to proposals, hopefully. And then there's 3 that I will go in a little bit more detail that really have to do with what was used to be our task. Our leaders is published data to the world. So AMBench is additive manufacturing benchmarks created by Lyle Levine at NIST. So they have all the data that they get from 3D printed materials or objects like the one in the top right. They do lots of different kinds of experiments and measurements on those and make those available. Then people who have models can actually try to predict those, so there are competitions for that. They wanted to have their data available to all their collaborators, but many of those are outside of NIST. They would have to get NIST accounts and all of that. Even though we have an instance of SciServer running at NIST, it's not as big as ours. It's running in the cloud, it's more expensive, well, arguably more expensive, so they bring their data to us. So people can run it here, so there's a demo notebook that generally with data, demo notebooks, how to access the data, etc. So these are run against the data that we have here. Another new data set from last year was by Gerard, one of the Hungarian students that often visit us. Millennium simulation is one that I used to work with a lot when I was still before here. Alex helped me set up a database with that. They run 2 different simulations, one with 1 power spectrum, and another with the same 48 modes, but then inverted amplitudes. So where you have a high density over density in one simulation, the same place with a lower density in the next one, so that's interesting. It's about 40 terabytes that's available. Other one is called Smudge. I don't remember what it stands for. This is individual galaxy simulation, only in dark matter, meaning gravity only. But then they have two, one with a bar, one pure spectral, spiral, one, and then for each of them also one where a dwarf galaxy will fall through. This is 154 terabytes in our system, but you can access it as it is local. It's not necessarily always fast, because it is a very large data set, depends on what you want. You can do all these analysis again. They provide demo notebooks that, for example, here on the right side give you orbits of stars in these galaxies. Okay, the new thing that's exciting is that we got money from the NSF to do something about the sustainability. It's a new thing, I think, for the NSF. They don't like to pay for sustainability, but here they did. And what we decided, or what we propose, is that we make SciServer completely open source, a proper open source project. Not just checking the GitHub repo to go from private to public, but we want to improve the code, the documentation, set up a strategy for how to deal with external people providing or proposing code updates and pull requests. That's one of the things. And we also want to make it easier to deploy new instances of SciServer in the cloud, for which it might be interesting for a workshop somewhere, a class somewhere, in some school. They set up a SciServer instance for themselves in the cloud to spin it up and use it that way. So these are deliverables, we'll start working on them. So there's a lot of developer documentation, Java code cleanup, making some things independent of Microsoft SQL Server, make it possible for open source databases, etc. There are going to be extensions for making it easier to set up a class in SciServer. There will be a workshop. We're collaborating with quite a group of people. This helps us getting the money as well. So people that we know from our projects, the online ones are these ones that have their own installation already. So this is one of those. And we're working together with the Science Gateway group that will help us. They have expertise how to do outreach, how to get people in to your project, how to set up a workshop, etc. That was it, pretty much. One other new thing that we did recently was we got our mugshots taken. Not everybody is here yet. It's okay to set this up. So now we have all our professional places that we put in LinkedIn, on our website. We haven't done it yet here, we haven't done it at SSEC. But so these are all the names of the people in our team that participate, not just in SciServer, but also the IT personnel, outreach people, database admins, etc. I think that was it. Yeah, that was it. Thank you.
So Kate's going to set up this talk. And I'll introduce Keith. Keith, you can come on up. Keith Clark. Is a PhD candidate in material science and engineering. He works on the A-Flow++ framework to discover new materials for clean and renewable energy applications with his advisor, Oses, who's here as well. I think he's going to get your talk up. Think of anybody's talk. Keith is flexible. Probably the one who says Keith. Scared. Yes. I'll give you a roll for it.
Alright, hello everyone. As I said, my name is Keith Clark. So I'm presenting
materials I can use, or that are possible. And so you do this for the binaries, for the ternaries, for the quaternaries, and you see that that number just it skyrockets, and you end up with 237 million. If you just have 6 different elements. And so that's why we're here at the data-intensive research center, not the experimental one, because that's way too many for experiments to handle. So our solution to this is to use high throughput calculations. So specifically, we're using the A12++ framework, as announced in my introduction, and we're developing that to generate these structures high throughput. And the way that we do that is we begin to generate structures to run with density functional data and think of material properties. What we start with is called a prototype. And so you can think of a prototype as, imagine you have some structure you've synthesized in the lab, you've done it experimentally, you characterize it, you've gotten the lattice parameters and all of these things, and you convert it into a geometry file. And this geometry file can then be used for you can run a calculation based on this geometry file alone, or you can extract its symmetry elements and all of the different symmetry operations that you can do based upon where the atoms sit, and you can apply those same elements and same constraints to a different system. Effectively, what you do is you take your structure and you extract out the backbone, and then you're effectively just left with a generic prototype of whatever structure you can. And the idea here is that you don't want to start with random combinations, because you could start with random combinations, but just statistically speaking, it's not very likely you'll end with real materials. And so if you begin with real materials, the idea is that you'll end with real materials. And obviously, we want real materials. We don't want to just base them on materials that don't exist. And so here are some other prototypes that already exist in the A12++ framework. You can see we have unaries, we have binaries and ternaries, and we have a lot of diversity in terms of the prototypes that we have. There's over 1,100 of these as well inside the framework. So our idea is that we prototype real sulfides, and so there's the inorganic crystal structure database with many different experimentally found materials. And so the idea is that we do the prototyping process on these materials, we extract the symmetry elements, and we can generate them and decorate them with different transition metals. So here I have silver sulfide, and as you can see, we can extract the prototype and then redecorate with different transition metals, whichever we choose. Of course, they have to make sense, and that's part of it. But the idea here is that one prototype can use many different structures to explore, because there's a wide range of chemical flexibility and all those things. And so what I've been working on is automating the sulfide prototyping process. And the idea here is that we start by getting all of the ICSD sulfides, all the experimental structures that we want real materials, so we start with real materials, and then we use the A-flow symmetry or A-flow sim computational pack module within the A-flow++ framework that was developed by Dr. David Hicks, one of my collaborators, and we use his software in the A-flow++ framework to find all the unique structures between all the ICSD sulfides that we have. And then we compare all of those structures to existing prototypes. If they exist, then that's perfect. We can decorate them already because we've already done all the symmetry analysis. If they don't exist, then we have to inject them into the A-flow++ framework. We have to inject them into the A-flow++ framework directly so that we can then decorate them and generate data using these new structures. And so that's pretty much the workflow. And then we calculate we generate the data on the materials properties. And overall, this will culminate in obtaining a sulfide training data set that we can then use machine learning on to extrapolate different properties, such as things for stability analysis and things like this. We can use machine learning to do that, and we will, and then we will use that. We will extrapolate the optimal compositions for the metal organic frameworks or for these batteries. So that's overall how it's been going. So far, a lot of the time and effort has been devoted into the software part of it, so actually getting this workflow down and being able to start with the directory of the structures, do all these imagery analysis, and then automate the injection into C++ framework. Here are all of the references that I have, and I just want to thank the team that I've been working with. So again, my PI, Professor Oses, the co-PI on the project, Professor Szalay, the history graduate students, and SRE is just the Institute for Energy Laboratory, which is the part that I'm in. And that's me, Keith Clark. Chen is also here, and I want to thank him. Hao is not here, but I still want to thank him. And the SRE undergraduate students are William Cyber, who you'll see in a couple of minutes. He's also speaking. Amy Cardiago, Gerard, and Nicola Brown. We're also new students and postdocs, so if you're interested, feel free to email us at jobs@s4e.ai. So that's the presentation. Thank you. Which cations we might decorate. We can prioritize certain ones over them, over other ones that we see added through literature search. Maybe it's been used for this other application that we think should be promising for this. So we can prioritize different transition metals. It's a very automatable thing.
So, sorry, you may not be able to answer this, but maybe you might have some insight into this. Why do phone batteries still suck?
Sorry?
Why do phone batteries still suck?
Oh, that's because they're not lithium sulfur. Once it's lithium sulfur, it'll be sulfur. Of course. Yeah. No, I do not know the answer.
So you alluded to them in the beginning.
Oh yes. Oh well, I'm not saying that they're terrible right now, but they'll become better with the lithium sulfur. I mean, all the benefits I listed at the beginning. They should be able to last longer. It'll be more lightweight. All of these things. So.
Other questions about? Yes. You talked about automation. I was wondering how you encode and inject the prototypes into the model.
Okay. So how do we inject the prototypes into the C++ framework? Okay. So I do that. So the way that I have it right now is that you can start with a directory of structures. If you're on your computer, you have a directory, and all of them are different geometry files and whatnot. It runs through the A+ symmetry code, which extracts all of the, which for a given structure, here's a slide. Here's a slide that kind of is more in depth about what a prototype actually is. And so the A+ symmetry code effectively determines two types of the able prototype, or not two types, two parts of the able prototype. There's first the label, and then the parameter set designation. And so when you run it through the A+ symmetry code to get the symmetry analysis, what you're really doing is you're first getting the able label, and that contains all of the symmetry operations. It effectively describes a set of symmetry operations that will that governs your system. And so you can see that there are different elements to it, there are different portions. You have the reduced word geometry, the Pearson symbol, space group number, and then this is a binary, for example, and so you have your write-off letters for your first species and your second species. And so that's all of the symmetry elements, and then on top of that you have your parameters, which these values, it seems like they're a little shifted in the bottom over here, but they're color-coded, so that's good. The A corresponds to the lattice parameter. The C over A corresponds to the ratio of the C value versus the lattice parameter. And then the Z1 and Z0 these values, so the vertical position of these atoms. And so your first part is really an infinite set of solutions constrained by so you run it through the prototype code, or the A-flow symmetry analysis code, and then what you have to do is, after you are able to that, you have all of your unique structures, and then or what I did is I have it so that it generates a large JSON-L, or a JSON file, or a file where every single line is a different JSON that contains all of this information about each prototype. And then it injects a new line if a new prototype needs to be added. So it'll add the new label, the new parameters, all of this symmetry information, and then it will convert the JSON file into C++, and it actually affects, it's recursive on the C++ code base itself, so it actually rewrites and overwrites one of the files that's in the code base. And then when we recompile it, we can automatically start using the new prototypes. Thank you.
We should move on to our next topic. So this brings us up to another one of our keynote speakers, Hartung. So Tom's going to work his way up, and Kate is going to bring us to the next presentation on the stack. I'm going to get in trouble if I do that.
You might do it quicker than that.
Oh, I think we'll do it quicker. I'll read part of the introduction instead, because I haven't met Tom before. But, Tom is the director of the Johns Hopkins Center for Alternatives to Animal Testing. He's also a chaired professor of environmental health and engineering, molecular microbiology and immunology. And has been working to steer the revolution in toxicology to move away from this 50-plus-year-old animal test to more organoid cultures and the use of artificial intelligence. So without further ado, Thomas Hartung.
Thank you very much. In biomedicine, we see at the moment two disruptive technologies which are changing very much the way we are addressing how we are modeling disease and how we are addressing, for example, the safety of substances, one of the areas I'm active in. AI is the one, this is the one which is bringing us together. The other one is that we are, by bioengineering, increasingly able to build organ substitutes of human cells, which are in many aspects superior to animal testing. I will show you first what brought us into AI, and then I will talk about organoids and how the combination of AI and organoids is leading us to organoid intelligence and what the possible consequences of this are. I made my slides available. I have the QR code also on the last slide if somebody is interested. As you can see, I listed here Frontiers in Artificial Intelligence because I'm the field chief editor of this journal. It's a journal which has, over the last 4 years, published about 800 articles in the field of AI in essentially all areas of use. I'm personally leading the area of medicine and public health, and we have 13 categories. So a little shameless plug for you if you're looking for a journal which is fully open access to publishing. Healthcare at the moment is, as any area, very much impacted by AI. I had the privilege, because of my role as editor, to write for the World Economic Forum the draft for the transformative map of healthcare by AI. If somebody is interested, I think it is really important to see that all of healthcare, this is about 17% of our GDP, is at the moment affected by the transformation through AI. Another shameless plug, we are at the moment preparing a Keystone Symposium with two of my co-hosts on AI and biomedicine, which will take place in May of next year as a virtual symposium. My main job here in Hopkins is I'm chair for evidence-based toxicology at the Bloomberg School, and there's highest expectation that also toxicology is benefiting from this. It is a remarkably lagging back in scientific innovation. I just turned 60 last month, and most of the tests we are using in toxicology have been introduced when I was not yet born or in kindergarten. So this is showing you how much we are relying on animal testing and have not really innovated. The expectations are clearly that we are able to replace some of these animal testing, for example, with AI, but also that we can dramatically accelerate. If I want to know for a given chemical whether it's a carcinogen, something which is, I think, a very justified request, it takes you 5 years to get the answer. It will cost 400 rats their life, and it is a $1 million exercise to find this information. Multiply this with 350,000 chemicals which have been registered in the 19 most developed countries. You can imagine this is mission impossible. We cannot use these methodologies. We would need something which is dramatically accelerated to get information on many of the things we are using. In the US, we are adding 4,500 chemicals to food. More than 80% have no adequate data to assess safety. So we are permanently consuming stuff which has not really been evaluated. So it is about accelerating this. And it's also about democratizing knowledge, democratizing between countries, but also democratizing with the various stakeholders. People would like to know about the things which we are using in our consumer products. I like the new toxicology, which is embracing AI, to call it toxicology. And this is becoming possible because we are transforming from a data-poor science to a data-rich science. Instead of counting dead animals or tumors in an organ, which you could do even without a handheld calculator, we are moving now to something which is based on high-content imaging, on digital pathology, on high-throughput testing by robots, by the various omics technologies, sensors, and also an enormous amount of literature. PubMed is our most important database. It gets 1 million new citations every year, and 80% of them describe some substance effects, not as necessarily as drugs or toxicants, but as inhibitors, something which is being used. So we gain enormous knowledge in this. At the same time, we all know computers are increasing in power, and this allows us altogether to make big sense. And this big sense in toxicology is, first of all, to find all of the information. There's analysis done that a typical risk assessment for a chemical uses only about 18% of the at the time available literature on the substance, because it is so difficult to just find this stuff. It is also about evidence integration. The more important, we need to make sense of various information sources, exactly the ones which I have been listing here. The more we use them, the more difficult it is to make sense of a combination of these. And we also want to predict. If you're interested, these are 2 articles about toxicology and AI, which will come out next week, actually, and I'm happy to send them if you're interested. Our major entry into the field was this study, which is using machine learning for toxicological data. Tom Lüchtefeld was at the time my PhD student. Toxicology is a pretty small area. This paper was downloaded 24,000 times in the first 2 months, and covered, the whole process was covered twice in Science and Nature. This is very unusual in this field. So what he was using is something which is called read-across. So if you don't know anything about a substance, you might know something about similar chemistry, and if you look around and read across these properties, you might save time and money by not testing and simply ascribe these properties. What Tom did do is he created a relatively large database and he made an automated read-across. And interestingly, this was capable of predicting 87% correctly the properties of 190,000 cases where we had a chemical and a classification based on animal tests. This compared favorably to the pure reproducibility of these animal tests, which is only 81%. So the machine was already, with this very early random forest-based type of machine learning, was already outperforming the animal tests. This was, for us, quite sensational. So by use, and this was possible because we were using not only the network effects of this read-across, but we also were using transfer learning from in total 74 properties. So, for example, as a toxicologist, I'm interested whether the rabbit eye is affected by the chemical, it's corrosive or irritant, and traditionally, computational methods would look, what do I know about other chemicals in the rabbit eye? But I might not know nothing about neighboring chemicals on rabbit eyes, but I know that this is a skin corrosive, or it is reactive chemistry. And then I can use this information to support my judgment. This is what the AI was doing. This has instant consequences for the replacing of animals. After this study, we showed that we can also predict human data, not just what the outcome of the animal test is. And again, it performed better than the best animal model in predicting whether a human would have a skin allergy in response to a chemical. A year ago, we did publish that we can also do this in batch. We took the 4,700 chemicals which are relevant in food in the US, and we did run them in less than an hour. We did the equivalent of 38,000 animal studies, which would cost more than $250 million if you only find the laboratories to run all of these. And again, we had about 83% predictive capacity, certainly about what animals would do. So you could run the entire chemical universe if you only wanted to. And we showed this year, this was only shown at conferences so far, not yet published, that we even can predict more complex endpoints like cancer or damage to an embryo with reasonable predictivity. So this is really something where we might be able to help to decide which substances smell like a problem and should be studied more in detail, where we can, on a massive scale, evaluate the backlog of testing. As I said, 350,000 chemicals registered for commercial use, and most of them never tested. We have the privilege that there is a EU project of $20 million, in which I'm leading the AI part, and the goal is to use the same methodology now for liver, kidney toxicity, and the developing brain, very important toxicities which are difficult to handle. The project is based on an interesting tool, which is based on machine learning. The first one, again, this is Tom Richtefeld's work. Tom has created during his PhD 2 AI companies, Topstrack and Insilica, which are located here in Bethesda, and he's still 50% research associate in my team. This program is a very interesting one, SysRef. It is allowing systematic reviews of the literature in a semi-automated way, where the machine learns which to include which articles from your first 100 or 200 selections, and then is going through the literature and is suggesting further articles to include. Besides this tool, which is really semi-automated and it's free for to use, and about 10,000 systematic reviews projects have been run on this platform already. We're using our tools, databases of historical data, and we use a meta crawler for information of the internet, the grey literature, which is available, which all of them feed data into our process. I found Tom's approach to databases especially remarkable. He's using something called BioBricks, which is a one-line command for importing databases within minutes. He has created, with support by NSF, the National Institute for Environmental Health Science and the European Union, 50 such BioBricks for the most important databases, which are going to be released shortly. Some of them are already on GitHub. The first 8 of them allowed us to combine to a database of 200 million cases where we have a chemical, a property, and a result. And this is already this is massive data in the field of toxicology. This is something really worth training on. What we're doing with this is now that we try to use chemical similarity, as in the previous approach, and biological similarity. This is illustrated here. These two are structurally similar, but not biologically. These are biologically similar. And by combining these two approaches, He is actually already showing in some test runs with just 1 million of the data points that he improves the prediction of 152 different properties for all of the chemicals, the 1 million chemicals, that he's upping the predictive capacity from about 70% to 80%, and we hope that we find the supercomputer power for running the 200 million chemical comparison, which is an incredible 130 quadrillion pairs of chemicals which we have to compare. If we run this, we hope that we further increase these potentials. So I think this gives you a bit of a flavor of predictive toxicology coming into reach on the basis of AI and the massive data which can be accumulated. The second technology which is really relevant is microphysiological systems, the bioengineering of cell cultures to produce not just a layer of cells which we keep alive, but really the architecture and the functionality of an organ. This is really a disruptive technology which became possible in essence with the availability of stem cells, human stem cells. And they became available as embryonic stem cells, highly discussed ethically in 1999, and in 2006 as induced pluripotent stem cells, as reprogrammed skin cells, with the famous invention by Yamanaka, and who received the Nobel Prize just 6 years later because of the fundamental importance of this in the life sciences. We have done a number of workshops and also published here a very nice review paper if you're interested in what these microphysiological systems are doing in the field. But we also created now, a year ago, the first World Summit for these microphysiological systems. The first one was in New Orleans last year. This year we brought 1,300 people together in Berlin. We are now moving to Seattle next year in order to discuss these numbers. My own area is the developing brain organoids. They were published first in 2013 by Lancaster's group, on the Nobel's group actually, by Lancaster as the first author in 2013. The same year we published the first time on brain organoids. In 2016, we then described how we can mass produce identical brain organoids which look like these perfect balls, identical in composition. We have been interested since in using them for autism research, for showing that COVID-19 is actually affecting human brain cells, or, for example, grafting them with glioblastoma. A very versatile tool, not the topic of today, but it is something which is really showing how in the life sciences we get relevant human models now from essentially any organ we would like to have. And increasingly, we are able to build disease models. In our case, this is for autism, for Alzheimer's, for some viral infections and others. So these are micropathophysicological systems. And what you might be able to see is here occasionally a flash. These are neurons, and every time they're depolarizing, there was an interaction between two of these neurons. This is calcium flows, which you see here. And when I saw this the first time, I said, wow, this is spontaneously occurring. I said, they are thinking. And then some people in the audience were shocked and said, they are thinking, are they conscious? And I very easily said, yes, but they have nothing to think about. They have no input and no output. And this prompted the idea that we might be able to do something giving them input and output. So while there is at the moment discussion that artificial intelligence could help us finally to talk with animals, there's a lot of projects in this area, we asked the question, can we talk to a brain organoid? Can we, by combining AI through electrophysiology, through microelectrode array, get to some type of communication between these systems? And we coined the term of organoid intelligence as a synthetic biological AI, reversing what computers have been doing for the last 60 years, or 70 years in the meantime, which is making computers more and more brain-like as as shown by artificial intelligence, by saying, can we make a brain organoid perhaps a bit more computer-like? And if you compare computer and brain, the brain is quite a remarkable computer. This is the Frontier computer, one of the leading supercomputers, which reached 1.1 exaflops in June of last year. This is the human brain, which is estimated at 1 exaflop of computational power. So only last year we exceeded what is the estimated computational potential. This is done, this required a 680 square meter installation for $600 million and used about 1 million times more energy than a human brain. Or you can say it in a different way, if you compare to a current laptop, which has about the size and weight of a brain, and uses about the same energy as a brain, it would take us about 33 years more of Moore's law to reach the same computational power in a laptop. Yeah? Just to illustrate this, and the energy consumption is obvious. So the idea of giving organoids by connection to AI some type of possibility to compute opens up for some visionary ideas. We might be able to do physiology of learning, the type of things we at the moment do by poking electrodes in macaques in order to understand how different neurons are working. We might be able to do in a human system. We might look into brain-machine interfaces, because here we have a human brain structure which we connect to electrodes. We can do TOPS and drug development by using disease models. If we develop a brain organoid from Alzheimer's patients, we might see how they are deficient, and they might be able to develop drugs around this. And we can dream of biological computing, where we use some of the superiority of the system. In the two-dimensional cultures of neurons already have been used occasionally for some type of control of systems. Quite prominently, collaborators of ours from Cortical Labs in Australia last year published on a system where 2D neurons were learning to play the computer game Pong. So this pedal here is controlled by the neural network, and it is improving in a training session and plays better and better, keeping the ball in the game. This is, I think, a very nice illustration by using this virtual embodiment, how the system is able to learn. Unfortunately, it forgets everything until the next day, because there's no long-term learning in the system, but it is just very easily explained because the machinery for long-term learning is not present in these structures. The paper was published with an editorial from our side, and we are working with the guys from Cortical Lab on, they're now using our brain organoids because we believe we have the machinery for long-term learning, and we are trying to do some similar work also here. So the three-dimensional structures providing the infrastructure, especially we have the glial cells, the helper cells, which are necessary to prune the connections between cells, which is the substrate for long-term learning. And we believe that we have actually evidence for long-term learning in our systems, and we are preparing for some publications in this field. It has also several other advantages. I don't need to go into details, but we have myelination, which is increasing conductivity of the transmission. Three-dimensional structure is 1,000 times denser than a two-dimensional culture, so we have much more connections between them. The discovery grant from Hopkins allowed us, with bioengineering, with David Garcia's group mainly, to develop a type of EEG for the brain organoids. These are self-folding electrodes. Here you see the graphics design. Here's the real brain organoid within these shell electrodes, and it allows us with very nice signal-noise ratios to record what is happening and then test, for example, the effect of a neurotransmitter which we have. We are running at the moment a Sampath project between the Writing School and the Applied Physics Laboratories. Eric Johnson is my partner in crime. We have a big ethics component with the Berman Institute for Bioethics, where we are producing similar virtual environments, but also implementations where we are trying to control robots, which shall take place early next year for the first time. We're trying to train these systems. So this is a very interesting addition in the discussion on intelligent systems, which has been enriched by the discussion how far artificial intelligence can be intelligent. We are trying now to bring into this organ intelligence, which could also be considered as a synthetic form of intelligence. You can imagine that this is producing a lot of ethical questions. This is an article in Nature already some years ago asking, can lab-grown brain spheres become conscious? So it is very important to us that we have a strong involvement of ethicists in our work. We call this embedded ethics. So they are planning with us our experiments, they come to the lab, they see these models. Currently we're working with systems which are about 500 micrometer in size. 50,000 neurons, that's about what a housefly has. So if they see that they see these are very humble approaches or very simple basic forms of learning. It is not a brain organoid which is talking to you. But at the moment we are working with the bioengineers already on 1 centimeter sized brain organoids. This is already twice a mouse brain. Then you can expect there could be really some type of intelligent behavior which could be produced. It is also opening up for a type of experimental ethics. We could ask what is an equivalent of intelligence in such a system, and then you can start looking how much does it take, how big has it to be, what are the components, what are the requirements for input in order to get any type of behavior which fulfills the criteria of intelligence in this system, not intelligence of human type. I'm very much interested to understand whether we can build disease models on this basis. We work with the Alzheimer Center here in Hopkins, mainly with Vassos Nachlaki. She has already models of stem cells from Alzheimer patients, and they show some of the typical features of Alzheimer histopathologically. What we now want to see is this also visible with respect to organ intelligence. So what we are creating at the moment is DORI, the Dementia AI Research Initiative, with some more colleagues, clinical colleagues also in the Alzheimer field, because we want to see, is this a way forward to study the disease and possible treatments. This is not the only disease. We also work on different new developmental diseases, autism associated, but also amyotrophic lateral sclerosis, autism, and some rare genetic diseases, leukodystrophies, which all impact on cognition. But this is not necessarily the end. It is not only about seeing the physiology and pathophysiology of a human. We could very well imagine that we could produce 10 kilogram sized brains. They're not distracted by all of the other tasks. We could use any type of architectures which we want to build. We can use completely unphysiological inputs like optogenetics, use training schemes which have nothing to do with physiology. So it is really a system, at least theoretically, it opens up for tons of opportunities. In February of this year, we published this idea as one of the inaugural papers for Frontiers new flagship journal, Frontiers in Science. We instantly made it into the White House Bold Biotechnology Goals as one of the goals to produce such an intelligent system. The National Science Foundation is going to announce a program on engineering organ intelligence probably in the next 2 weeks, from what I hear. So there's something interesting happening. A number of editorials were published around this, a lay people version, a children version, and a couple of activities. So it is a very much Hopkins-centered approach. You see probably some familiar faces like Alex here, who helped us with a lot of aspects for a large infrastructure. But we also have external partners. partners which are working with us. We carried out the first workshop, which led to the Baltimore Declaration towards Organ Intelligence. The workshop report has been published. It is part of the philanthropic goals of the writing school to build up some competence centers here. And in closing, there's nothing new under the sun. This is Star Trek from 1966, the second season. You see this computer based on brains which Captain Kirk is discovering. And so already then they were dreaming of a brain-based biological type of computer. So I hope I showed you the revolution which is taking place with respect to models in biomedicine. Toxicology, the use of AI is obvious one example, but also organ intelligence as a vision at the moment, more than actually already delivered results, but something which is coming into reach as a possible complement to the in silico type of AI that we are using. Thank you very much.
Questions for the speaker?
I don't know if this is a fair question for you, but what do you see as the barriers for the organization of animal testing?
Honestly, mostly it is change management. AI and also these organic models are in many instances at least equivalent. The problem is to get so many stakeholders who have been using the old methodologies for 60, 70 years to get them to change. This is also associated with aspects of liability. Is this defendable in court? When there's a liability case, can you defend that you have not done an animal experiment and there's possible damage? How do you show that science has moved forward? But it is, I think, the most important, there's two ways of moving this forward. One is to move it away from saying this is for ethical reasons, but by demonstrating the economic consequences and needs. The second one is to make this a co-pilot, not switch completely the system. To say, yeah, okay, we have now the opportunity with AI to condense information, make predictions, you learn how good this is, and after a while, you might change after some optimization steps. This is like when we changed from horse coaches to cars, we did not change from one day by dropping one technology, but we learned. They coexisted, and at some point, it simply made more sense to drive a car. So I think we have to change, and that's my main.
It's not just the ethics of animal suffering, but it's humans who are psychologically scarred by doing animal testing for years and years.
It is there's a lot involved. It is humans, as you rightly say, are suffering. We are also suffering from the misleading results of animal tests. Many treatments which would have helped humans have been sorted out by misleading animal experiments. And we are wasting enormous amounts of money because we are using tests, or we're using this machinery which is delaying also, for example, drug development by years. And we just had a wonderful example. COVID-19, all of these vaccines and the antivirals have been developed essentially without animal testing. And they were enormously successful. Normally 6% of vaccine projects lead to a marketed product. In the case of COVID, we had nine vaccines, all of them successful to some extent, so that they came to the market within 1 year, and this essentially without animal testing.
One more question in the back.
Yeah, this is also maybe related to ethics of different kinds of the toxicology when you're doing this artificial intelligence and doing the tests, and then presumably to check that you need real cases from hospitals to see whether what you predicted is correct. And is there a system in place for getting that information so you know whether your predictions are correct? And how does that aspect of it work? That's one of the big problems. Yeah? We can either use the traditional data in animals as to calibrate our methods, but then we stay with the limitations of the animal tests. And I cannot on the one hand say these are dreadful, and then I say we use them to benchmark. The way out here is, from my point of view, mechanism. You typically don't get enough data from humans being intoxicated so that you could take results from this, but you can ask, are mechanisms triggered which we associate with disease manifestations, and this is where things are converging, and where my hope is that toxicology actually will be changing from the science of poisoning a rat to the science of understanding what chemical exposure contributes to disease. Human exposome, which is like the human genome, addressing systematically what chemicals do. There's enormous potential in doing this. Mechanism is the clue to bring it together. Great. Just a quick follow-up. Is there epidemiological studies that would follow up on this to understand that? I mean, can it be done in conjunction with these toxicology initiatives?
There is some, now with biobanking, large biomonitoring studies, we start to get information, and exposomics, the application of different omics technologies, brings them down to some mechanistic proposals. But it took us 50 years to show by epidemiology that smoking is producing lung cancer. It is not an approach which has high throughput and is leading us very quickly. But it makes a contribution, but it's not the solution.
Thank our speaker once again. So for our next speaker, we have William Scheiber, who's going to talk about one of the summer fellowships. Has a slide ready to go.
This summer I worked to develop and implement A-Flux at JHU, which is a material search API for the A-Flow data repositories generated in atmosphere optics. As Keith was mentioning earlier, A-Flow is an open source high throughput framework for materials, calculations, and simulations. And Engineering for Energy works to develop A-Flow to model materials for clean and renewable energy, which carries a volume of new data, which prior to the summer we had no way to access. So the old implementation of A-Flux was written in PHP and was not integrated within the A-Flow framework. So A-Flux at JHU is written in C++ and is built directly in the publicly accessible A-Flow framework. There were some additional. So that basically eases the use of A-Flow and gathering data, so you can access the data and contribute your own data more easily to the A-Flow infrastructure, which is why we say we created this federated system as different universities can easily input their data and access other universities' data while maintaining data provenance. A-Flux at JHU included some other additional improvements to the original A-Flux implementation, mainly being increased filterability. We added tensor-like filterability and species and atom-specific filterability. And so now we're allowed to query and filter on materials and tensor or species-specific properties. So querying is basically just requesting information from the database. So at the prior implementation of A-Flux, you could query on, say, a specific tensor for a specific material and get that back. It was impossible to filter on that. And so, but those tensors are valuable information when you're doing calculations for science. So, for example, the shear modulus is an important property of a material. And you can calculate that using the law of approximation, which takes in a couple of components of the stiffness tensor, which is calculated for that material. And so it's easy to see why you might want to be able to filter on those specific components. To do this, we added a new operator kind of to the A+ language, which is this @ symbol, which basically allows you to define where you want to splice and filter on in that matrix. Now, the more difficult part was adding the species and atom-specific filterability because of the dynamic nature of these vectors. We use a relational database, and every material has a different number of species and a different number of elements, and a different number of atoms. So all these things were different lengths and different sizes, so it's very difficult, practically feasible to filter on using SQL in a relational database. So to solve that, what we did was create a second layer of filtering in C++, again, still directly within the A-Flow framework, that allows users to then filter on these species-specific properties. An example of one of these properties is, like, say, the Vader atomic charge, which the Vader charge is essentially the formal charge of the atom. So it's easy to see why you might want to be able to filter and search for materials that have specific charges and specific atoms within their structure. So then, up on the screen here, see I've got a little flow chart of essentially how the process works. You know, you get your A+ query in the A+ language, just a bunch of different parameters and directives and everything. We then translate it to an SQL command and then send off to whatever DB file or database you want to query. That searches and filters the data there, and it's brought back to A+, which then goes and is brought through the secondary filtering, which is in C++. And finally, that data is packaged as either JSON or an HTML file, and it returns to the user for machine learning or whatever they want to use it for. And then up there, there's an example query and some of the data that it might return. So I'll translate this. It's basically searching our database for materials that contain both iron and oxygen with a greater than 0 density and less than, and a beta charge at oxygen of less than 0, with at most 3 atoms. And then the last two things are just directives. So the paging just returns the first 25 and then the format in HTML, more visually pleasing than looking at a JSON file. So that means you can access the data and filter on the specific materials that you might want to use. And then we've connected this, made this more, because we've integrated it within the directly within the A-Flow framework, it's more plug and play. So any other universities using A-Flow or research centers or whatever that use the A-Flow framework can easily access the data and also distribute their own data, enhancing material science research efficiency. Thank you.
who's an associate professor of economics here in the JHU Carey School of Business. His work focuses on social and strategic interactions affecting individual and aggregate socioeconomic outcomes.
Well, thanks. First, thanks to the IDES for this grant. It was useful. We hired 2 research assistants that are helping us with this project. Copier wanted to be here, but he has a small kid. He lives in France, so it was a little bit difficult to make it. You know, I hope I can deliver. So this project, systemic risk and externality software dependency network, is a mouthful, but if you look at the picture, you more or less understand the motivation. So we started thinking about how software is developed nowadays. So despite what movies would like us to think, there's not one person in a basement in a dark room working on his computer and developing some packages, but this is more a team effort. At the same time, modern software developers use a lot of code that already exists. So you may create this functions that call some other function in other libraries. Why do you do that? There are some pros. Well, and there is a documented increase in efficiency of developing code during the years because of reusing existing code. There are some cons, and that's what we're trying to focus here, is that once you create a dependency to another package, you increase the risk that there's some bugs that is not yet found in that package, or there is some vulnerability that is going to be discovered later, and you're basically increasing the risk of having some vulnerability in your package. Why do we care? As economists, and as you know, Copier is an expert in financial contagion, because this is actually a big deal in terms of cost. And so the cost is estimated to be over $1 trillion from all these software failures. And this includes vulnerabilities as well as bugs and disruption in delivering performing code. And the number of incidents that were reported increased at least 10 times from the beginning of 2000 to more recent years. So this is actually a problem that is relatively large in terms of business cost, and is also an increasing problem. And so what we do in this project, and we have also some follow-up project, but in this project we're trying to model the decision of maintainers to create these dependencies. And the idea is that these maintainers will weigh cost and benefit of creating all the code in-house or using some functions that call some other libraries. At the same time, they may partially consider some of this risk, and so we have a way to model how they consider this risk. We model also the fact that these maintainers may have different quality and abilities to develop code. We analyze a model that has all these ingredients, and we analyze the equilibrium of the model, and we can estimate the cost, the benefits, and all these externality of risks. And then, since Copier is an expert of financial contagion, we have a way to measure the systemic risk for the ecosystem of software. To give you an idea of what the data look like, so this is, each dot here represents a package, a library. The ecosystem here is Rust. Not because we think Rust is, very important or the most important language, but because it was, kind of manageable from the point of view of our estimation as a first application. And so each dot represents a library. An arrow from one dot to another represents a dependency. Okay. This is the kind of data that we're looking at. In particular, if you look at this kind of network, this is a dynamic evolution of 3 libraries, A, B, and C. Each library has is a version that includes 3 numbers: the major, the minor, and the patch. And I'm being very sloppy here, so if there are computer scientists in the room, I hope I'm not offending anyone. But essentially what happens is when the library is created, it's at version 0, right? The major is 0, but then it gets updated. If the update is minor, it's just like a few updates, then it's a patch. If it's relatively larger, several patches, it's a minor update. And if it's a revision of of the code or addition of many more functions or many more routines, then it becomes a major update. And so here you can see the evolution of this network of 3 libraries, where we have A, B, and C that are connected. And so A uses functions and routines from the B library and the C library in the second iteration, but then once it's updated, it just needs to lead directly to B and not to C. And so this is the kind of graph that we observe in our data. We call it a dependency graph. It's been studied a lot in computer science, as you can imagine. So it's not new. The data are available online, so you can download all this from libraries.io. So far we have downloaded Rust, we have Python, we have R, we have JavaScript, and there are some other languages that you can play with. We also have some measure of activities of the maintainer, so we know, how many times they release new versions. We know how long these packages have been available, how many stars they get in GitHub, and how big is the package in terms of bytes. We can also do a number of lines of codes. But just to give you an idea of what kind of data we have. And so we have a model. I'm going to spare the mathematics. If you want to see the technical model, you can just read the paper. I can point to you at the paper. But essentially, we have one maintainer that makes decision about creating this dependency. Okay, so every now and then, over time, the package needs an update. And so the coders propose either we update in-house or we create a link to another function in another library. And so the maintainer has to make a decision. He has some cost of doing this, right? Creating code in-house is going to cost. Creating code that calls another library is going to cost a different amount. And then some benefits of doing each of the options. The other thing that he's going to consider, he may look at this library that he's trying to link to and say, okay, let me monitor this library for several bugs or several vulnerabilities that I may find. He may also go one step further and say, okay, this library itself has dependencies, so let me look at the libraries that this library depends on, and you can keep iterating on this process. So you can go away from this library, one link away, 2 links away, 3 links away in the network. And so that gives us a way to test how far the maintainer is willing to pay the cost to examine these libraries. Once the decision is made, the network is updated, and we go to the next field, and this process starts again. Okay, so over time, basically, we have a network that evolves, and these dependencies change over time. We can show in our model that this converges to a stationary state that gives us the likelihood of observing a particular network in the data. And so we estimate that the parameters of the model using maximum likelihood. Now the issue is that the likelihood of this model is what we call a mixture of exponential random graphs. And so there are some challenges that we have to address in terms of estimating the model. So these exponential random graphs, if you've done statistical mechanics, you probably have seen these are likelihood with retractable constants. So it's impractical or basically infeasible to compute the likelihood or derivatives of the likelihood. Simulation methods are very slow for these kind of models. And the size of the network here, since the network, for example, for us we have 17,000 nodes, is going to make things worse. And we can show that the number of simulation steps is proportional to the square of the number of nodes times some constant. So how do we get around this challenge? We use aberration approximations, minorization methods, spectral methods, maximum zero likelihood. Usually people choose one of these methods and approximate the likelihood using one of these methods. What we do is we basically use them all. And we have shown in some simulations that this technology, this machinery, actually doesn't compromise accuracy, especially in large networks. So we also developed a package. So this was in the works for other work that I've done. So we developed a package. It's available on GitHub. We are releasing it on CRAN very soon. And so this basically allows you to do the scalable estimation that we do in the paper. So you can play with it. There were some packages that will allow you to do this before. We have improved the speed by sometimes 14,000 times, which is a good achievement. We have decreased the amount of memory that you need to use these packages. For example, the estimates for Rust that we have in the paper as a preliminary result only use 20 gigabytes of RAM. So a standard desktop can do this. If you have, if you have a larger network like JavaScript is 2.2 million nodes, then you may require a little bit more RAM, but you can do this. I'm not going to spend too much time on the results because we're working now on a new version, so these results may change a little bit, but we find evidence that these maintainers inspect the dependencies at least one link away. So they look at the dependency package, they look at their dependencies at least one link away. Sorry? I'm almost done. And the other thing that we see is there's some evidence that these maintainers compete for attention in the market for open source. And so they have some competition going on among maintainers and among packages. There is some evidence that they tend to link to libraries of maintainers or packages that are similar in terms of quality, both unobserved and observed, and in terms of activity, in terms of maturity of the packages. Now, what we're doing now, after we received the grant, the first thing we did is we're actually working on the code to make it even faster or to solve some of the memory problems. We are analyzing other languages. As I said, we have collected data for JavaScript, for Python, for R, so we can redo the analysis for anything else. So basically, we develop a pipeline to go from the data to the estimation to spit out a table with estimates. And so we can replicate this with whatever data set we give it. Now, the additional part is we're adding a model of contagion. So as I said, Copier has studied financial contagion during the financial crisis. We have these models in finance of how banks interact and how banks lend into each other, increase the systemic risk, depending on the structure of the network, the topology of the network. So here, given the network of dependencies, we can do some similar analysis. We can borrow from the literature and do some similar analysis and give a number that's basically it's a synthesis of the systemic risk imposed on the ecosystem of software. And then given the estimated model, we can simulate multiple networks and give basically a distribution of systemic risk, so we can give you confidence bands on that systemic risk estimate. And then, in the next few months, we're also tackling the dynamics. All the analysis that I talked about basically takes the network at a single point in time and does the analysis for that particular network. But ideally, there is a dynamic component here, as I show you in the slide. We have changing libraries, updates in the libraries, and they usually create problems to other libraries. And so we want to analyze also that dynamics. And so this is all we've been doing for the last few months. Thank you.
We have time for a few questions.
Yeah.
How do you integrate the costing models into this analysis?
Sorry, I integrate?
How do you integrate the economic costing model into this?
We have maintainers that have some payoff function that they try to maximize. The payoff has some cost and some benefit. That is a function of observable and unobservable characteristics. Now, of course, we can also, we're trying to tie these parameters that we estimate to salaries. And so we're collecting some data, outside this library that I have, we're collecting some data on salaries of software developers. A lot of these projects are open source, so there's not necessarily a salary, but we can, sort of like give a monetary value of that cost. Yeah. It's mostly model, I would say. Yep.
Could you elaborate a little bit on how you developed that initial dependency graph for Rust? Like assumptions made about the size or usage of a particular package?
The size or the usage?
How did you get that initial dependency graph for Rust? Like I can just write some Rust code in it and get it, right?
Yes. So we go on libraries.io there is a package manager, and we just get the data from there.
So that would be like Git for Python or something?
Yes, pretty much. Copier would be more precise about this, because that's his part of the paper.
Curious about your thoughts about the parts of the system you have visibility into, things that Combariza may want to have or just want to have versus lots of development languages in a closed setting that you may or may not get access to. How different do you think that will be?
We don't know how portable this is, because this is mostly open source. I'm not sure.
It's dark, you don't know what's in it. Any other questions? Let's thank our speaker once again.
Thank you.
I'll introduce the next speaker. This is Julia Lane. Julia is a professor at NYU, economics. In her bio here, she's won lots of awards in lots of societies. I know Julia since last year, 1.5 years ago. When we started our SSEC project here that I talked about this morning, we got the choice of some projects as a kind of proof of concept that we started working on before hiring RSEs. So we worked on it with IDES personnel, in particular, some of them, you see the names there. And there were 3 projects, and this one seemed to fit really well with the skill sets and the work that we have been doing already at IDES for a long time. And it has worked out really perfectly, I think. Started working on databases, pipelines, web interfaces, machine learning. Streams Futures doesn't want you to continue a project once you get to a maintenance phase. The nice thing was that Julia started providing other sources of money from the government foundation, federal agencies such as NCSES, USDA, NOAA, etc. So, in a sense, sustainability has been her coming up with money. She's also come up with new ideas, with new projects, and really the only person I can compare her with also for her willingness to travel all over the world is Alex. I thought that he was unique, but apparently in economics there's more people like him as well. So I'm very curious to hear what you've been working on this last 1.5 years. Sure, yeah.
Okay, so I'm not going to stand up there and talk, so I'm going to walk around and pick on Gerard if I can. Turns out the secret to working with Gerard, if you've ever worked with him, is you just constantly feed him salted licorice, or promise to feed him salted licorice, and then everything's okay. Okay, so this really has been a fun project, and actually I was just at Ohio State on the next part of another project that's funded by the NSF's Technology Innovation Partnership Director, which is, I'm a measurement guy, so it's about measuring AI. And I'll talk a little bit about that in just a minute. So I'm going to give you a bit of context about this particular project, which Gerard talked about, and then give you an idea of what's been done and what else is going on. Okay, so I've spent my career working with large-scale, quite complicated data sets. I know that every field thinks they have the most complicated data set. We, in economics, social science, ours are very complicated because they're based on human beings, and so there's confidentiality restrictions as well. But here are the basic challenges that we face, I think. I came late because I drove in from Pittsburgh today, so I missed a lot of the conversations. But the problem that we have in working with data is there's a zillion different decisions that are made at every stage of the way. And because no data set is ever clean. And what you really want to do is you want to have lots of people looking at it. But the provision of that research data to a broader community is lacking, right? So all the returns come to the individual. They don't, even though it's a public good, it's publicly funded, there are lots of disincentives for sharing the information. And I don't need to go through that. I've been on a zillion NSF panels, and I was an NSF program officer at every stage, in every directorate, every field that I've been in. The disincentives for sharing research data are massive. So here's the issue. We've got this, it is to everyone's benefit if these research data are shared and reused, but it doesn't happen very much. Next slide. So what we really should be thinking about, and as you know, as I said, I'm an economist, how do we set up an incentive structure so that it is in everyone's self-interest to share the data? So you go to any kind of conference and everyone's saying, oh, we're trying to make it easier for people to share data. 20 years, and that equilibrium is suboptimal. And the problem is, it's just that there isn't a market. Right? So Alexis pointed this out a long time ago. You don't get bread and so on from the butcher and the baker and the brewer because they're trying to help out the public good. They do it for their own self-interest. And here's the issue, if you move it forward. I was told very early in my career that there are 2 units of academic currency: grants and publications. That's what matters. So if those are the units of academic currency, if you hoard the data and you have monopoly power over it, then you're going to get the most grants and the most publications. So what we're going to try and do in this project, and the reason that our buddy Stu Feldman, Alex and I got to know him, well, you knew him further back than I did, but a while back, but the reason that Stu and others from the Foundation and NSF funded this is to try and think about how we can democratize that approach. And I was struck by the fact that the preceding couple of speakers talked about it. So next slide. So here are some thoughts. Keep going. Oh, so what we really want to do is to think about, instead of brewer and baker, how can we think about creating incentives for the researcher, for the publishers, and for the research institutions and the research agency. So think about a research agency or any federal agency that's collecting data on human beings. There's only downside risk to making data publicly available because, particularly when it's confidential data, if there's a breach of confidentiality, everything blows up. So they also, for all the reasons that Gerard knows, they don't allow the information out. So what we started to do was to think through how we can do this. So next. Because if you don't align incentives, if you don't make the market system work, and you just do what the science agencies have been doing and say, you will have a data management plan, or you will put data on the web, or like Nature, you'll provide the underlying data pieces. It'll be like the Soviet Union, where you have a quota system. You tell people that you have to produce a nail by volume. You get one big nail. If you say it's by quantity, you get lots of little nails. So trying to manage it top-down bureaucratically is the wrong incentive system. So it's got predictable results. So keep going. And we saw that with the push in, I was at NSF in 2009, with the Obama administration, the Hartung memo came out and it said, we're going to have open data, we're going to make data available, we're going to force agencies, we're going to force people to do this. And what we got was a vomit of data. We're in the same situation now. We have a Hartung memo in 2012, 2013, Now we've got the Lemson memo saying the same thing, but without the right infrastructure put in place, we're going to get into a mess. So next slide. So the advantage that we have now that we didn't have back in the day is we have a push for data to be made use of to produce evidence. And I noticed again one of the previous speakers says, we want to go from data to evidence. And so both in the Foundations of Evidence-Based Policymaking Act and in the Chips and Science Act, next slide, they're being told, they have every federal agency, so it's the 24 CFO agencies, that they have been told they have to post information about the usage of their public data assets on their website. So they're getting a bit more explicit than they did back in 2009 with the open data. So they have to post information about the usage. Now, all of a sudden, we now have the research agencies and the programmatic agencies that have to post how data are being used, not just put a vomit of data out, they have to say how is it being used. Now, how are they going to do that? So suppose you're a federal agency, so I'm going to pick on someone. Okay, that's why I'm walking around. I'm going to pick on you. So you're a federal agency, you're Health and Human Services. How are you going to show how your data are being used? They created chief data officers for each agency, so the monkey's on their back. How are you going to show it's going to be used? Or NIH? How are you going to get a database of who use it?
Have the researchers report their use of the data.
Have the researchers report their use of the data. How well do you think that's going to go?
It's usual, we're getting asked more and more things.
Yeah, you're getting asked more and more things. And you might say how you've used it, but if you make it available, how are you going to figure out how that's being used? And how are you even going to collect that information?
Well, if we had DOIs for the data, then we can track it.
If you have DOIs for the data, good point, right? So, for the past 15 years, the agencies, or the science agencies have said, let's have DOIs for the data, and then magic's going to happen. So they've had this huge effort, data science and all of these kinds of things. So let's follow that thought through. Next slide. I was on the National AI Research Resources Task Force. We have the same kind of issue. If you take a look at this, keep going. We worried a lot. When I got appointed to the task force, I was telling Alex, the first thing that happened was Jaime called me and said, we need more GPUs. Spend the money on GPUs. And I'm like, look, mate, we need better data too. How are we going to get better data? There's vast amounts of data out there. How on earth are we going to figure out that of the quality of the data and the usage of those data? Because you could spend, you just heard in the earlier presentations, you could spend the entire GDP of this country on curating data sets. Ain't going to happen. So we've got to figure out how to build a community around data. We've got to create incentives for people to do that. And what we argued, of course I was on the net, so you'll see that part of what I've been doing here with IDIES is building a search and discovery portal, because if I can find the data and then I can track it, then I'm going to automatically get usage statistics, and that's going to set up the right incentives, because the researcher is going to get credit for the data that they produce getting downloaded, just like as you were heading with a publication, the agencies are going to get credit, the publishers can provide that as a service to reviewers. So it's all incentively patterned. So we could just automatically generate those usage statistics and highlight them and reward people, then maybe we could think about 3 units of academic currency: publications, grants, and data assets. Right? So that's what we're going to do. And we've got the legislation and the proposed legislation in place. So we could set up a search and discovery portal for the NAIR, if it's ever funded, if Congress ever elects a speaker, but those are 2 other things. And then that could be used, so if I'm searching for a particular topic and I want to find out what data sets there are, people upvote it, I'm going to get more credit for it, and it automatically curates. So it's like an academic currency, like the Hotchkiss, the Baker mechanism. Right? Next slide. So all through that report, which is what we submitted to President and Congress, and I'm going to say it's not as well written as it should be. There were 12 of us on the panel. I was the only social scientist, the rest were computer scientists. It was great fun, just let me tell you. It was a joke, yes. I know I'm standing between you and drinks. Next slide. So what we said in this is strengthen AI data resource and the infrastructure. Next slide. And if you take a look at, I'm sure you'll be sharing these slides, we have a whole bunch of things on what should be done around it. Okay, go. Same thing with this research data framework. They've got a whole set of recommendations. I think a lot of you know Bob Hamish, so he's been leading this, and so it's all congruent with also with the NIST standards. Okay, so what's been done? Next slide. You said put in DOIs. So our buddies at NASA, they actually went through. They have DOIs for their data. This is one of their data sets. They went through and pulled 100 publications to see how many of the researchers cited their DOIs, and out of 100, 18. And it took them 14 hours to go through and manually figure this out. So providing DOIs is kind of like telling your kid, how many of you have kids? Not sold on the local market yet? Save a lot on college tuition if you sell them first. But then, so like telling your kids, I've got 3, so it's, Ian, go clean your room. Ian, go clean your room. It does a really bad job of it. What I found out is 2.5, put gold stickers up, and he'd clean his room. If at the end of the week he had 7 gold stickers, he could get some money and go and buy candy away. So think of researchers as being truculent 2-year-olds. Let's figure out how to give them stickers, because there's no advantage to putting this up. So this is where IDIES comes in. What happens is these bloody researchers don't cite the DOIs. What they do is even the data sets they created, like Scimetrics, that Superwoman, that's me, did not cite the data set. So how are we going to find it? Well, it's hidden in plain sight. This is a machine learning problem. I'm going to go through and I'm going to say, how are researchers naming their data sets? I can pull out the data set names. I then have a publication data set diag. Once we hook the publishers in, we can just suggest a DOI, suggest putting it in the references so it becomes a virtuous cycle. At least in the interim, we're going to find those data sets. We're going to look for them. Go to the next slide. And we're going to start with the federal agencies, the most touched by the Evidence Act, which is statistical agencies like the National Center for Science and Engineering Statistics. Next slide. And then what we're going to do is we're going to build the equivalent of an Amazon.com for data. So what we have now is a situation just like a bookstore 30 years ago, where everything's manual. What we all recommended was manual. So it's like going to a bookstore and saying, well, I want a book like on X. And if there was someone at the bookstore, they'd say, well, here it is. we can automate this process, build an Amazon.com-like website, and have the community rate the quality of the data and so on, and upload what works and what doesn't. So next slide. So you can write little reviews and stuff. Next slide. So once you've got this, the whole idea of this being, because the Evidence Act has really had such an impact on the federal government, this is the kind of theory of change, that's what economists think about, is how is this going to help them do their job better? So this is the agencies that we've been working with have identified these as series of change, and they include NIH as well. You know, the input's going to be data products. We're going to go and get the identify the data sets. We're going to get immediate outputs like the APIs and the dashboards that we built with IDIES. And then that's going to the outcomes that we're going to be looking at. And if you take a look at the NAIR report, we talk about those outcomes, better research productivity, because you're not wasting so much time replicating people's work, and obviously get more congressional interest, because it's an underfunded resource in general. Okay? So next slide. So what are we going to do? We're going to use machine learning, natural language processing models to find data sets of interest. We're going to get that data set publication diet. Once we've got the publications, we know the topics, we know the citations, we know the institutions, we know the authors, we know the regions, we've got some sense of size. Does that make sense? So that's what we've been doing. And we've been really successful. Next slide. You want to go and take a look at democratizingdata.ai. That's our initial stat of a website. Keep going. You can take a look at the user guide, which describes the approach that we've taken. Keep going. And effectively here, this describes the process. Johns Hopkins has been very big at this side of the piece, of how to develop the metadata, the data model, and then feed it into the API. Keep going. So, what happens is agency will identify its data sets. This happens to be a list of data set names. Obviously, you're also looking for aliases as well. Next slide. Sometimes they're a little bit noisier. They're looking not just as data sets, but they're looking at the data assets that they produce. Keep going. And that then gives us the incentive structure that I talked about with the usage statistics. So we're able to show who are the authors, who are the top authors using the data sets. And if you don't think that's an incentive structure for JHU and for the researchers to say, I am the #1 researcher that has produced this data set in this topic. I'm the world expert, right? So now you're starting to think about data sets becoming an actual asset. The agencies can highlight how their data sets are being used, and this is automatically generated. I don't have to manually do something. You know, so you think about, I think the one guy said, we've been doing animal experiments since before he was born. It's the same methodology. So we've been effectively using the same methodology to describe data usage. We don't have to. We can automate it and develop a self-reinforcing mechanism. Next slide. And the agencies and the publishers and so on are getting right into it. Okay, so next slide. Keep going. Oh, that's just going to show a video. We're not going to have time, so keep going. So what are the next steps? So we've shown that this can be done. Again, thank you, IDIES. Now the next step is to make it a two-way conversation, just like Oses did with Amazon. So the agencies are starting to fund the workshops. Keep going. This is one example. They wanted us to go to Kansas City, and we did. Anyway, it was great fun. Very good barbecue in Kansas City, it turns out. Keep going. We've been engaging the community through the Jupyter notebooks. So, like, the challenge with publications is all of those usage statistics are done behind closed doors. General impact factors are generated. There's no transparency. With the SciServer, the community gets to figure out what those usage statistics could be. And they can be domain specific. Keep going. We're building training classes because you don't just want to have a few people working with the data. You want to explain how the data can get used to go to evidence. Next slide. Find out not just the scientific impact, but how it's getting picked up in Wikipedia and social media, things and Twitter, or X, whatever it is. Next slide. So, to close out, research data is largely a public good. What we've been doing is starting to think through and how to build it out, not just from the benevolence of the brewer and the baker, creating those incentives, keep going, and create 3 units of academic currency. So that the data assets is in everyone's self-interest, because we're not going to be able to do decent AI if we don't have decent data sets, and if we don't deal with the incentive structures, we're just going to get overwhelmed with, well, politely put, a vomit of useless information. So that's the idea behind and the recommendations as part of the National AI Research Resources Task Force that I was on. Part of what we recommended, that was the other White House committee I was on, was the advisory committee to the evidence building. So now is the time to do things better and develop a market, an academic market around producing data. Okay? So that's the end of the story. And I'm on time. I didn't even get to people who are on. Yeah.
So if the aim isn't for controlling or scaling data, right? So like you got a mechanism for incentive.
I know. Yeah.
You have
well,
so you have a mechanism for incentivizing data production, right?
And data curation. Because the usage is only going to occur if the data are well curated. If it's bad data, it cannot be used.
My counterexample to that is, have you searched for anything on Amazon recently?
Yeah, so he mentioned my interview about research. Amazon turns out to have become an integrated example because all they're doing is ads, right?
Oh, that's not actually what I meant. I meant the spamming of low quality products in every class.
Yeah, so it turns out Amazon is not the best example. So you want a highly curated academic style piece. But the original idea of Amazon, which was to get the community to upload things, like you've got, and Airbnb's become degraded. So I'm not saying it's a perfect idea, but the core notion of a community rewards for providing good information and getting upvoted is the underlying idea. And I'm sure we could do a lot better than Amazon.
Well, I mean, I'm just talking about the cost side, right? Because like the way you can actually implement that is by having like the cost of publishing, right?
So the basic idea here is, right now, there's only downside risk to providing research data, because people are going to come and whine and say, you didn't document this, and it's just a burden. So the basic idea here is, by putting this information up on the agency website, so that there can be controls about who uploads things, there can be community barrier or restrictions. And so I think it gets less and less spent. Yeah. The cost is getting downvoted and the benefit is getting upvoted if you do a really good job. So, you know, part of the thing here is I built the ADHD program at the Census Bureau, which was the first, still the only national statistical infrastructure that was built by a researcher. And it turns out there have been thousands of articles that have been written using this. And obviously I don't get credit for it. So it'd be nuts to do something like that. So the basic idea here is building an infrastructure where the person who does that gets the rewards. Yeah. So there may be better ways of doing it when Amazon.com is just kind of using it to fix something. But the point's a really good one. How do we think about how to do that? There's a lot of good economists who can think about how to do that.
So interesting, last week we were talking to Lynn Parker about the same report.
Sorry, we were talking to?
Lynn Parker.
Oh yeah, Lynn at Tennessee. Yeah, she was
Yes.
co-chair at the beginning.
Yeah, so she didn't address that in the same way that you're doing that. But we were discussing that, and you mentioned this, funding agencies like NSF, they have mandates.
Yeah,
But
every agency.
But they don't fully enforce that mandate.
That's right.
When you write the final report, you can say, I did this to disseminate my data, to share my data, but they don't care. Probably read the report, but they don't go and check if that is true. And they don't care about quantity. They don't care about how many people share your data. And that is part of the problem, that there is a mandate, but they don't have the workforce to enforce that.
So I'm going to share a dirty little secret as a former NSF program officers. Most program officers don't read the reports at all. I had a colleague who tried this out.
Don't do that, I spend months.
But what does work, you know, you think about computer science, you'll see in the computer and information science and engineering panels, the computer scientists will have how often their code's been downloaded. They'll put that on their CVs, and they'll say how many followers, and they'll put their GitHub by the month, because GitHub reports it. And on that, like, we ran the first edition of this, we did a casual competition. And they have masters, like grandmasters, they have levels of expertise, and part of that is also not just did they win the casual competition, but are they recognized as a master in sharing code, and is their code, are their notebooks really good and reusable and so on. So there's a remarkable amount of sharing associated with that. So my point is if you try and do top-down, where you've got, Ian, did you clean your room? I don't know your first name, but let me say, Gerard, did you write a decent report? It's not a very effective methodology. But if you have a community that is upvoting you because you did a really nice job on your own data set, and you can put that down. I wrote a piece in Nature, and I'm very fond of citing that. It's the 5th ranked of the Nature articles of the same age, and it's got 107,000 downloads. I put that in my report, and I cite that in what goes in front of the panel. That's the kind of incentive infrastructure that we'd like to build around data assets, and it's got to start around finding out who's used the data. Right? I can say there have been 107,000 downloads or 200 citations because that's automatically tracked. Create that same infrastructure and data sets and code. Does that make sense? So I think that's the challenge we've got to face. We've started the conversation. I'm not saying it's perfect, but I think that's where the attention has to be paid.
Thank you, yeah, real interesting. Yeah, I shudder at the thought of NSF having sort of enforcement mechanisms. I much more like the sticker idea.
Yeah. It worked with my 2.5-year-old. I'm certain it would work with people like you.
One thing you didn't mention or talk about, I was wondering what your thoughts are in terms of the resources needed for longevity of curation of data sets, because there's sort of a, it's a sort of with a paper or research grant, it's sort of a one-shot thing. You write the paper and then you're done, or you get the grant and then you finish the grant after 3 years, you put it on your CV. But very often these data sets, you create them and then you have to keep maintaining them for longer than the typical sort of grant. And so what are the incentives of resources?
This is Alexis' argument, right? So maybe you could say a few things about that, but he talks, about how much it's being used, but that's not the automatically track. I think if you did something like this, you'd probably get 10 times, and then the reuse is potentially massive because it'll you'll figure out how much it is continued to be reused, and that then is an argument for continued funding of the infrastructure.
I think the funding agencies will need to be sensitized to proposers making that argument. My data is being used this much, you can be funding for 2 more years to keep maintaining the data set. I think that's
and they are. So we've got NIH. So part of the advantage of the Evidence Act is there's chief data officers in every agency now, and because they're being asked to put the usage of their data assets on the website, otherwise we'd just be talking to each other. But that is the killer thing for us, right? Okay. And that's why they've been funding us. He says it's because of Michael Brooks' brains and personality. It ain't. It's because of the Evidence Act. We had a call here.
Yeah, so I guess a similar question, what Gerard was saying here is that how do you kind of prevent this from being abused? Or not maybe not abused, but obviously being dystopian if you have these metrics for certain data sets that maybe some groups are very good at kind of hacking those metrics to make their data set look more useful, and then the funding agency using that to determine where the grant money goes. Whereas you might have a very small data set or a data set that's used by a small group of people that maybe is very valuable for what it produces, but that metric won't capture that.
So a couple of responses on that. I'm going to push back on the use of the pronoun. How are we going to do it, not how am I going to do it, right? So what I'm saying is, this is not a problem for Julia Lane to solve. It is a common issue, right? So I'm going to push back on that. How do you stop abuse? And I've got a couple of responses to that. One is, there are some isolate data sets that might not have a lot of use, but are unique. And Tyler Christensen, who's at NOAA, her husband works for the Forestry Service, and they have high-valued conservation decisions, again, based on, how important is it in an ecosystem? Is it a unique type of... So the Forestry Service has identified a set of different measures. I don't think we should just think of one measure. And part of why SciServer has been so useful is having communities figure out what's the highest value thing. Now, in a market, it's easier because, you've got a price, but I think that's our community decision. It is critical that those usage statistics, not just counts that the agency puts up, and that we have some say in it, and so that's the bottom up thing. So I'm putting this to you as the beginning of an important but in charrette area. Yep.
Right, so I just want to ask, is this plugged into the sort of big data, small data sets, things that are out there now? So you have Webb telescope data where one thing collects a giant data set and everybody uses it, or there's other things like that. But you also have things like cryo-EM or the structural biology side of things where, well, that will collect a pretty large data set. Maybe it's uploaded, but think about how we're going to store this and who's going to pay for it, sort of thing. They upload the final result of structure, you can track sort of that. Maybe, I don't know if it's plugged into like the PDB or EMDB or these other sites that host this data. We don't keep the raw data forever because
it costs.
Can't.
Yeah, so part of the deal here, and what we're working with NIH with the Office of Data Science Strategy, is starting with some highly obvious NIH data sets, get that community figuring out what the value proposition is. You know, they do have these heavily funded data repositories, or federally supported data repositories, and they have to make some decisions about what to keep or not to keep. And so, you know, people talk about the calculus of curation. Right? So something that calculus is going to have to be made with evidence about the contribution of the data sets, and some are going to need to get sunset if you're going to not be eaten by a giant data monster, right? So we need evidence to make those decisions. So, yep.
I was going to suggest stopping here,
Shut up.
because Alex still wants to close this meeting, then we can go for drinks, and no doubt Julia will be there.
And by the way, I'm just going to say, I'm in New Zealand tomorrow at 3 o'clock US time. The All Blacks are playing the Irish in the Rugby World Cup. So if you're watching it, you know, say a few prayers for the All Blacks.
Thank you very much for this wonderful energetic talk, and also for reminding us in this age when we are rushing madly forward to buy more and more GPUs on the importance of data.
Yes.
Thank you very much. And I think this is also kind of a good way to stop. So on this kind of a perfect closing remark that really what Julia delivered. So putting everything in balance and in context, why are we doing all this, so that we can make better decisions everywhere in society. So closing this, I would really like to thank everyone for coming, and I would like to thank the people who actually made this happen. So thank you very much for Kate, Elliot, and so for everyone. And given the sharp upward trajectory that we are, I think this repeat of this meeting next year will be probably even more exciting, and hopefully we can find a tropical